Dr. Kapoor?
Yes, Professor Del. Yes.
Will I be able to see the slides on my screen as they're shown to the group?
I believe so, yes.
Okay. Just wanted to.
Just like yesterday, what we tried, that should work that way.
Okay.
कोई ऑफिशियल ऐसा नहीं है, it is chair. Or when we say we can start. Okay. Can we start? Nile, are you recording? Let's start in. Okay. A very good morning, good afternoon, and a good evening to all of you. Welcome to the NECLIME conference organized by BSIP Lucknow, India. Yesterday we had a very interactive session. Had 10 talks yesterday, and the chair was Professor Torsten. Today we have 2 sessions in total for today, and 14 talks. And in the first session, the chair was Dr. Vandana Prasad, but due to her commitments, urgent commitments, now the session will be chaired. The first session will be chaired by Dr. Amit Ghosh. So, Dr. Amit Ghosh, sir, please.
So very good morning in Germany, and good afternoon in India and other parts of the world, as well as in different parts of the world like US. It is a different time. So thank you very much for joining in this online NECLIME meeting again. And today, in fact, Dr. Vandana Prasad is preoccupied due to some urgent meeting. And on her behalf, I am giving you the proxy. The proxy is a very usual term in paleontological studies. So I am giving the proxy instead of your Professor Vandana Prasad. So welcome to the afternoon session. And the session will start first with a keynote address by Professor Daryl P. Domning. And the paper is authored by Professor Domning, Professor Sunil Bajpayee, and Dr. Vivesh Veer Kapoor. The title of the keynote address is Biotic Evolution and Biogeography during the Neogene in South Asia, Serenians and Seagrasses. So may I request Professor Dominic to start your lecture?
Thank you very much.
Hi,
If we're
good
going to
morning,
talk
everyone.
about the Neogene fauna and flora of South Asia, we certainly need to include the waters of the Indian Ocean, and that means talking about Serenians and seagrasses. Next slide, please. Serenians, also called sea cows, are air-breathing aquatic mammals, such as this manatee, who is proving she's a mammal by nursing her calf. Next. Today, Serenians are no longer as diverse as they used to be. We now have only 3 species of manatees, all living in the Atlantic region, 1 species of dugong living throughout the Indo-Pacific realm, and then there's stellar sea cow, this monster at the bottom, up to about 10 meters long, which did live recently in the North Pacific, but was exterminated by us humans in the 18th century. Next. The mammalian order Serenia has a fossil record going back at least to the Eocene. And for all of that time, over 50 million years, Serenians have mostly been tropical and subtropical, and mostly marine animals. Next. Serenians are also herbivores, and for the ones living in salt water, that means, with few exceptions, feeding on seagrasses, which are marine angiosperms as opposed to algae. This also means Serenians tend to be bottom feeders, especially this dugong, which has a strongly downturned snout, helping it to ingest these very small seagrasses almost like a vacuum cleaner. Next. As for their origins, Serenians evidently evolved from land mammals back in the Eocene and quite likely in South Asia, nearly at the same time and maybe in the same general region as the whales did. But they very promptly spread throughout the Tethyan Seaway and beyond, as far as the Americas and the Caribbean. Next. By the Neogene, say the middle Miocene is shown here, at any given time, there were numerous species of Serenians spread throughout all these 4 parts of the tropics. If you click on the slide again. And 3 more times, please.
Back.
Oh, no.
Yes.
That didn't work. Okay. Today I'll be focusing on the Indian Ocean, which region 4 there, with some mention of region 3, the former Tethyan and modern Mediterranean region. Next. I'd also like to talk about the fossil records of seagrasses, but unfortunately I can't because there hardly are any. So if you do happen to find any seagrass fossils, they're definitely worth collecting. But I think I can make my point just by showing you their modern distribution. As you can see, the Indo-Pacific realm, certainly including India, today has the greatest diversity of seagrasses found anywhere in the world. Admittedly, that means only 14 or 15 species at the most in any one area out of only some 60 species worldwide. We're not talking about the diversity you'd find in a tropical rainforest. But it's still a very nice salad bar for the dugongs to enjoy, including ones with small, medium, and large leaves and rhizomes. Next. Next slide, please. To explain what that means, we need to know some basic anatomy of seagrasses themselves. Here's a typical one. It has 4 basic parts: leaves, shoots, rhizomes, and roots. Most important for our present purpose are those underground rhizomes, which serve to store nutrients. and therefore are also the parts of most interest to the Serenians. In fact, something like half of the biomass of a seagrass meadow is in the rhizomes. So if you're not eating those...
I just want to interrupt you. Someone is has done a wrong operation. I'm just requesting her to, in fact, remove her presentation. Please hold on. Please hold on.
Okay.
Please hold on.
Present again. Yes.
Okay. Can we go again?
Yeah.
Okay. So, something like half the biomass of a seagrass meadow is in the rhizomes. So if you're not eating those, you're missing out on a lot. This species has rhizomes about 2 to 3 millimeters thick, which I would call a medium size for rhizomes. Next, please. Okay, here's a photo of a different species with rhizomes up to 1 centimeter thick, which I call large. Now that size may not impress you, but if you tried to dig rhizomes like this out of sand with your bare hands, you would quickly start looking around for a knife or some other tool. Or if you were a Serenian attempting it with your mouth, you'd want to evolve some large, sharp tusks. Next. Now let's take a quick look at Serenian classification. The order comprises 4 families, the first 2 of them confined to the Eocene. The third one is the manatees, and all the ones we'll be seeing today are in the dugongid family, which has 3 subfamilies, only 2 of which, the ones in purple, are represented in the Indo-Pacific part of the world. So keep your eyes on the Haligorinae and especially the Dugonginae. Next. Now, before I introduce these fossil species to you individually, I must call your attention to particular parts of their anatomy that give us clues to what they ate and how they minimize competition among sympatric species, which is to say ecomorphological characters. The single most informative clue to an animal's ecology is generally thought to be its body size. But since we rarely find complete fossil skeletons, we use the length of the skull as a proxy for overall size. Second, we take the angle of downturning of the bony rostrum or snout as an indicator of where in the water column they tended to feed. As much as 75 degrees or more downturning for an extreme bottom feeder, considerably less in others. Third, almost all dugongids have a pair of upper tusks, which we presume to have been used for uprooting seagrasses, especially those nutritious rhizomes. The tusks may be small and more or less round in cross-section, or longer, with the root extending half the length of the rostrum, or longer still, the entire length of the rostrum and flattened, with a self-sharpening cutting edge along its rear. In this extreme case, figure C, the whole apparatus resembles a box cutter, almost all handle inside the bone, with a short but sharp blade, making it a very effective harvesting tool. Next, please. This is how I think the larger, more blade-like tusks were used for cutting thick rhizomes. This figure on your left is a tusk with a sort of lozenge-shaped cross-section. The tusk has a thin layer of enamel only on the medial side. The rest of the tusk is made of much softer dentine, which wears away on the lateral side, leaving the sharpened enamel. The animal opens its mouth, as you see on the right, bracing the front of the lower jaw against the substrate, as shown at A, Then the jaw muscles at B contract, pulling the tusks with their sharp rear edges at C backward through the mat of buried rhizomes and allowing the rhizomes to be ingested. However, this action exerts an upward bending force on the entire rostrum, thereby generating a compressive force at D, which is the contact between the premaxilla and frontal bones. Different large tusked Serenian species adapted to this compressive force in different ways, in all cases by evolving some sort of butt joint between these bones there at D, which is evidence that the tusks were used in this fashion. Next, please. Here's an example of that using a species we'll talk about more later on. It shows the butt joints between the premaxillae, which form the rostrum or snout on your left, and the brain case on your right. In this case, they contact the nasal bones much more than the frontals, which are marked FR. But the effect is the same, to meet that compressive force head on rather than with a sloping plane of overlap between the bones. Next. Now let's meet some of the characters in our story. Here's a very typical Miocene dugong, a Hamadryene to be precise, by the name of Metaxatherium. This particular species is from the USA, but it's almost identical to the European one I'll be mentioning in the next slide. You can see it has a moderately downturned snout. It also has a very small tusk that you probably can't see. I would call this animal an ecological generalist, not as extreme in its feeding adaptations as some, but able to eat almost any form of seagrass. And it was a very successful genus of Serenians, being found in all 4 of those tropical regions I showed before, and surviving from the early Miocene through the Pliocene. Next. Now let's move back to the Old World in the Mediterranean region in the early Miocene. Note that at that time, the Mediterranean was still connected to the Indian Ocean. Later in the Miocene, that connection was blocked. The most common Serenian there and then was a Hamadryene species called Metaxatherium crahuletzi, found in much of Europe, as the purple arrows indicate. You can click it once more to bring up the arrows.
Click them.
There we are. And likely also in Libya in North Africa. Later it evolved into other species of Metaxatherium that had larger tusks. But at this time, at least in Libya where the blue star is, it coexisted with a dugongine, different subfamily, remember, named Rhytiotus heli. Now the zoogeography starts to get interesting. Rhytiotus heli has recently been identified in Madagascar also. On the subfamily level, this was no great surprise because until then, all the known Indian Ocean dugongids were dugongines. But this posed the puzzle of why there were no Hamadryenes at all east of Suez. After all, as the cosmopolitan generalists they were, they should have been everywhere. But that problem has now been solved since just 2 years ago, evidence of a Metaxatherium, probably crahuletzi, has also been found in Madagascar. Next, please. Now allow me to present Rhytiotus heli himself, a very impressive beast with a strong snout deflection of about 62 degrees and a pair of very intimidating self-sharpening tusks that could dig out even the toughest, most deeply buried seagrass rhizomes. So Rhytiotus and Metaxatherium, the specialist and the generalist, could have coexisted very comfortably in whichever ocean they both occupied. Next. Next we come at last to India, specifically the district of Kutch, which has produced nearly all of the good Serenian fossils in the Indo-Pacific realm. The animal on the left is dated to the late Oligocene, so we'll skip to the other one, which is Miocene, Baratisiren kutchensis. This is a dugongine similar in size to Rhytiotus, but with a rostral deflection of only 35 rather than 62 degrees. And tusks not as flat and blade-like, so it presumably had a distinctly different diet from Rhytiotus. Next. This one, however, Domningia sodai, is uncomfortably similar to Rhytiotus in its feeding equipment, almost identical rostral deflection of about 60 degrees and a flattened blade-like tusk. It may be wondered whether their feeding niches were so much alike that Rhytiotus and Domningia did not share the same habitat, one in India, the other in Madagascar. This hypothesis predicts that they will not be found in the same locales. Next. Another dugongine from Kutch, Kutchisiren cylindrica, is obviously different in its ecology from all of the above. Its rostral deflection is extreme, 78 degrees, even more than a modern dugong's, which is about 70 degrees. And its tusks have a lozenge or kite-shaped cross-section, evidently with enamel only on their medial surfaces, forming self-sharpening posteroventral cutting edges. These three essentially sympatric genera, Kutchisiren, Domningia, and Baratisiren, may have been pushing the limits of ecological coexistence. So it'll be interesting to see whether future collecting reveals still other competitors in their niche space, or whether they really lived all together at the same time. They may not have. Next slide, please. Perhaps the most tantalizing Serenian specimen to emerge in this part of the world is this isolated left maxilla, or upper jawbone, from Madagascar. The two figures on the left, which bears the Latinized Malagasy name Norosiren zazavavendrano. The ventral view on the left shows sockets for the upper cheek teeth. It's the dorsal view in the middle that shows what's unique about this bone. A low, broad, transverse ridge, abbreviated TR, lies across the path of important nerves and vessels supplying the side of the face. No other known Serenian has a structure anything like this, with one exception. Here on the right, a dugongine named Xenosiren yucateca from, of all places, the Yucatan Peninsula of Mexico. This beast is apparently later and certainly more derived than Norosiren. The transverse ridge is high and thin rather than low and broad, and it may have helped brace the cheekbones during vigorous digging with the tusks. Is this a case of convergent evolution, or a sign of true phylogenetic relationship between these species half a world apart? It's too soon to say. Next. Now it's time to add up the score. India and Madagascar are not so far apart geographically as Serenians swim. Surely close enough to share dugong species in common as they do today. But Madagascar has so far produced one Hamadryine and two dugongines, whereas India has three dugongines, but not a single Hamadryine, with no genera or species at all in common between India and Madagascar. This surely reflects just how incomplete the Serenian fossil record remains. The three nominal taxa at the bottom, by the way, are too fragmentary to identify with confidence or say anything useful about, so we'll just ignore those for the time being. From this list of herbivores and what we know of their morphology, what can we infer about their feeding niches and possible competition? Let's make a first crude attempt in the next slide. Assuming that first, modern Indo-Pacific seagrass genera are representative of the Miocene flora. Second, that seagrass rhizomes are an important resource. And third, that Serenians need large tusks for harvesting large rhizomes efficiently. We can then plot those variables, tusk and rhizome size, on a horizontal axis. At the bottom, we have the modern genera of Indo-Pacific seagrasses grouped by diameter of rhizomes, from small to very large. The vertical axis shows rostral deflection, which is a measure of degree of reliance on bottom feeding, whether on leaves or both leaves and rhizomes. Disconcertingly, this implies that most of these Miocene sea cows wanted to eat the largest and least diverse seagrasses because we see them clustered in the upper right-hand corner, corresponding to just the two seagrass genera in the large and very large rhizome categories, Thalassia and Halus. Two of these herbivores, Rhytiotus and Domningia, even seem to have overlapping preferences, implying serious competition. Meanwhile, most of the total niche space and presumed plant biomass is left to Metaxilarium alone. How to explain this? Well, perhaps not all of these Serenians coexisted in the same part of the Miocene or the same part of the Indian Ocean. Or perhaps other variables such as body size need to be factored in. Or perhaps the largest seagrasses were much more abundant then, or the seagrass flora was significantly different from today's in other ways. But this at least is a point from which we can start asking such questions. Next slide, please. Here's such a question. What about the modern dugong? Where would it fit in this scheme? We go back one slide, please. Just for fun, I've plotted it here. We go back. Yeah, there in the upper left-hand corner, I've plotted the modern dugong just to see where it fit in. And I've put it where the diagram shows it has no tusks at all. Well, as we all know, dugongs today have quite substantial tusks. But in females, they usually don't erupt. And the males don't seem to use them for feeding much, if at all. So for practical purposes, their tusks don't enter into this discussion. And both sexes bottom feed, largely on the smallest seagrasses, which don't really require tusks to dig up. So to that extent, this scheme seems to fit the available data. Now let's go to the next one with a nice male dugong skull with nice fully erupted tusks. In the next slide, please. Those tusks extend through nearly the whole length of the rostrum. Now, I have not addressed here the question of where this sole modern survivor of the once diverse dugong subfamily came from. Conceivably, it may have evolved from one of the Indian Miocene dugongs we just looked at, but I've so far detected no sign of which one that might be, nor is there any post-Miocene fossil record of any dugongs in the Indian Ocean realm that might allow us to trace its ancestry backward. Next slide. And yet, I have seen a late Pliocene specimen that would make an ideal ancestor for our modern dugong. Here it is, and once again, its origin is a shocker. Florida. It's a new genus and species, and in fact, it's the latest known dugongid in the whole West Atlantic and Caribbean region. It could have dispersed from the Caribbean to the Indian Ocean by way of the Cape of Good Hope, crossing the Atlantic at the same time as manatees crossed from South America to Africa. Why has it not yet been published and named? Because it's in a private collection. Next slide, please. Next slide.
That's it.
Oh, okay. All right. Yeah, you can go back to that one. There is much still to be discovered in the fossil record. To sum up here, and the Indo-Pacific region is the least well sampled of all. Even so, we already have an intriguingly diverse assortment of taxa from there, displaying adaptations for diverse feeding niches and likely reflecting a complexly structured seagrass ecosystem. In the absence of a usable fossil record of the seagrasses themselves, our best chance of reconstructing aspects of their evolutionary history may lie with the Serenians that fed on them. And the first, most general such lesson the Serenians have to teach us is this. Seagrass biologists must recognize that throughout the Cenozoic, most seagrass biomass has not just rotted and been recycled through the detritus pathway, as they have traditionally assumed, given how few living species there are. Rather, the norm has been for a very large fraction of seagrass primary production to pass through the guts of large herds of diverse, large, warm-blooded herbivores. This throws a very different light on seagrass ecology and provides an excellent example of how paleontology of animals can illuminate our understanding of the plants they evolved with. Thank you.
So thank you, Professor Domning, for the excellent representation of your work on biotic evolution and biogeography during the Neogene in South Asia, Serenians and Seagrasses. Basically, you told about the distribution in time and space regarding the dugongs, and also discussed about the Helitherini as well as dugongi. So I now request the participants, if you have any questions after his keynote address, you can ask, raising your hands by other means. So you are most welcome to ask if you have any queries, because we have enough time. We have at least 14 minutes time for that. Yes, I can see. There is a raising hand by Christine. Well, welcome,
Yes,
Christine. Yes.
Thank you very much for that fantastic presentation. I enjoyed it a lot. I'm not a mammal I'm not a marine mammal specialist, so it was very, very interesting. But I'm studying terrestrial herbivores, mammal herbivores. And the analogies between both of the ecological communities might go a step further. So I have two questions.
Okay.
My first question would be, would it be possible, in view of the potentially coexisting taxa in Kutch, the coexisting species, would it be possible to have like an ecological sequence, for instance, like in ecosystems in the Serengeti, so that different species would specialize on a different part of the seagrass? Or are they just targeting for rhizomes?
Well, almost anything is possible given the scarcity of the fossils of the sea cows and especially the seagrasses. I don't know if they would specialize on different parts of the seagrass. Well, to the extent that there were rhizome feeders, I think, and ones that would specialize on the leaves, different Serenians. Of course, any Serenian that could excavate the rhizomes could and presumably did eat the leaves as well. But then there would have been specialists, I think, on rhizomes that were accessing a resource that the ones without the large tusks would not have had access to. So to that extent, yeah, there would have been a division, I'm sure. But we really need a more comprehensive picture of the Serenian diversity. We're not likely to get much of a direct fossil record of the seagrasses, although that could change if we get into a different facies, different preservation, limestone or something like that, because there's a site in Florida, for example, Eocene site that does produce a diversity of fossil seagrasses. So it's possible in some places at least to get that kind of information. But as the record of both the plants and the animals continues to improve, I think the picture will get more and more interesting.
Yes, thanks a lot. And
you had another question too?
Yes, my second question would be, do we know anything about the mobility patterns of sea cows?
Well,
so that,
go ahead.
To explain, to get a clue for their strange, do they perform migrations, for instance? Or are they just staying in one spot where they find their preferred seagrass species?
Yeah, they can migrate, especially in the more temperate climates where they can, like on the east coast of the U.S., where in the summertime they can move farther north along the coast from Florida, which is their only winter range. Some individuals in recent time have been known to travel up as far as Cape Cod in Massachusetts, all along the East Coast, and then back again back to the south as the weather gets colder. So they can certainly do that, and they are capable of making long distance treks or swims, and they have dispersed widely in the whole Indo-Pacific region, out to some of the Southwest Pacific islands that are rather isolated, and so they are quite capable of traveling long distances. Yeah, they when they find a spot they like, they'll certainly stay there, but they can go quite long distances as well, which is why I was puzzled, still am puzzled, as to why there's no taxonomic overlap between Madagascar and India. There certainly is no barrier there that would have, especially with India farther south in the Miocene, it would have been closer to Madagascar. So there's an open question there as well.
Thanks a lot.
Sure.
I think Dr. VP Mishra has some question. Dr. VP Mishra, you can raise your question, please.
Thanks, Dr. Domling, for this interesting presentation on Serenians. As far as Kutch Serenians are concerned, I initiated work on it about 5 decades back with Professor Sahni, and now later Sunil Bajpayee and others continued it. We got these dugongings and what you termed as letter, not Metazoitherium or even Halitherium. The one mandibular MS I did record initially as belonging to Halitherium from the Oligocene exposures, but perhaps it has been redesignated to dugongings only. So some more data is required from Oligocene of Kutch, probably, it seems, because those are and regarding the Madagascar and the Kutch, as far as concerned, you say that yes, we don't have any common forms. We had to have some. We have to see some precursors of these Miocene forms, both at Madagascar and even at Kutch. Because during Eocene and Oligocene, it was a bit closer to the Madagascar. So we have to have some more data. And one thing I saw in the Kutch region while working there, it was long back, about 4.5 decades back, a lot of crabs, smaller crabs were found closer to the these Serenian remains. I don't know what was the reason. Why so many of crabs, small crabs, very small sized crabs, where the fossils of those were found close to these Serenian remains? Please, Dr. Domling, please.
Crabs I haven't looked at. We didn't find any in the parts of Kutch where I was working. I don't know if
I was. It was in Mata Numad, area called Mata Numad.
Yeah, we worked there. Dr. Kapoor maybe can enlighten us about that as to the crabs.
Yeah.
The part which is south of Mata Numad, if I can just make a submission here, like the exact age even for the Serenian yielding unit and the other yielding unit is bit of a thing, because it's either the lower part of the Khari Nadi formation, or it's either latest Oligocene or earliest Miocene. We do get crabs and other forms, but not directly associated with the Serenian specimens, where Dr. Dominic is right. And you are right that in that same region, crabs are formed, but either in the Eocene part, I have seen it in Ratonala and all those places, as well as in the younger Miocene ones, but not directly associated with the units which have yielded those Serenian specimens. So you are both correct in your own observations. Yes.
Maybe.
Is there any other question? We have again 4 minutes time.
I have one.
It's your paper. It's your paper. You should not ask.
Okay. Just a curious discussion.
I have one question. May I ask Professor Domling? I have one question. Yes.
Sure.
In fact, the food habit of your dugong, it shows that there are 4 different types of seagrasses you showed in your presentation. In fact, and their rhizome size are different and other things. But the question comes from Christina's query regarding the migration of the dugongs. You see that basically their feeding strategies are like this, that they were dependent upon seagrasses. Okay, so seagrasses normally thrive in the photic zone, not in the deeper part of the ocean. So how these forms migrated from one part of the ocean to the other part of the ocean, that is my question. Either because they were not able to go into the deeper part of the ocean, they were restricted in the shallower part of the ocean, that is in the photic zone itself, where the grasses are growing, because because of their food habit. So they should have migrated in the shallower part of the ocean, from one ocean to the other ocean, say for instance from Madagascar to Sri Lanka or whatever it may be. Okay, that is my question. So is whether from ecological perspective it can be resolved?
Yes, seagrass dispersal is another interesting question. There are, I imagine, vegetative parts or seed-bearing parts of the plants that can break off and travel with ocean currents, and also you could the seagrasses could disperse over time along coasts from around East Africa and Madagascar,
Yes.
around
Yes.
the coastline to India.
Yeah.
But we know that seagrasses are found on rather isolated islands as well, so they do manage to get across over time, and they have geologic time to work with. So that is a question. A question that you can take to the map of seagrass distribution and see where a given species is found and see how much isolation there may be between present parts of the range. The depth is an interesting question as well. Yes, they are shallow water plants, but they do, where you have very clear waters, get down to over 30 meters of depth, something like that, which is deeper than you would expect a Serenian, which are also shallow water animals, to dive. But in fact, it was found on the coast of Australia around the Great Barrier Reef that there were large areas of seagrasses, small ones like Haemophila, that were not known to exist because they were in the deeper waters. No one thought they would grow that far down, 100 feet or more, until it was observed that there were dugongs out there offshore in these deeper waters where dugongs were not expected to be. And then they went down and looked, and sure enough, there were seagrasses on the bottom in this very deep, for Serenians and seagrasses, very deep waters. And the seagrasses would not have been discovered there if it weren't for observing the dugongs. So sometimes these critters will surprise us, turning up in places and the plants as well that we don't expect.
Yes, that's true. So, okay. Thanks, Professor Domning, for the nice presentation and for the excellent presentation you delivered in the keynote address. And thanks again. And,
thank you for inviting me.
Should we move to the next presentation? That is by Grzegorz Worowik, and he's from Poland, I think. He's from Poland. He will present his paper on fossil calymothaloid fungi from India.
Good morning, good evening, and good afternoon. I would like to present my presentation, and now I would like to start it. Yes.
Professor Worowik, we want to see you again. Professor Worowik, can you for a minute you can just put on your camera?
I cannot start camera. I don't know reason.
Okay, no issue.
And I would like to start.
You now present.
Yes, sorry. Yes. Can you see it? Can you see my
not now.
Yes.
It's coming now.
It's okay?
It's okay. You please do the full screen.
Yes.
Please have your full screen.
It's full screen.
This is not probably the full screen. In the bottom, there are 3 icons on the right hand side.
Now? It's okay?
No. No.
Once more. Can you see my presentation on the small or full screen?
Huh?
Now it's okay?
Next to 110 on the left.
Next to 110, on the left hand side, you just see there is a funnel shaped icon.
Okay, yes, once more moment, once more. F5,
Shortly,
yes.
F4. F5. If you can press F5, that will bring it to full screen.
F5.
F5, okay.
F5 basically. Sure. It's coming back again. He's there.
Professor Borovic, your presentation is not here. Can you please start again?
Alt F5 है ना?
कुछ और दबा दिया होगा, मीटिंग ही लिख कर गया। चलने दो, ऐसे ही चलने दो, क्या करोगे? Your voice is not coming. Unmute. Please unmute. Professor Borovic, you are not audible.
म्यूट से भी हटाना होगा शायद।
Please unmute. Please unmute. In the bottom, you will find there is one icon.
Yes. Can I make a presentation in this state? Because when I push F5 in this presentation, it throw me out from this, so I can.
Okay, no issue. You please go ahead. Please go ahead.
Sorry for these problems. I would like to present my speech. Maybe I will do once more. This one and this one will be wider. Okay, it will be better now, I think.
Yes.
I would like to present my presentation about fossil kalimotaloid fungi from India. Next slide, please. Wow, how we can do it? How?
Arrow key.
Oh yes.
Yes.
Can you see?
It's coming. Yes.
Okay.
Yes.
Yes. F5. I don't know reason. Epiphyllous kalimotaloid fungi. There are epiphyllous fungi with sporodochial non-osteolite conidiomata with a single pore. It is conidiogenous locus aperture. The fossil one is Kalimotallus, most known, and modern one, for as example, is Myocopron letarere. It is you can see both fossil and modern has pores.
Microtherese.
Yes, there are examples of fossil kalimotaloid fungi from genus Kalimotallus, different forms. All are there are you can see that all forms have pores inside cells or in external part. Next one. The commonly recorded fossil kalimotaloid fungus Kalimotallus pertussus, previously thought to be related to fungi family Meteriac or some modern algae, Phecopeltis or Uvula, was found as almost identical to the modern Mycoleptodiscus disciformis and Neoecoleptodiscus venezuelense. To accommodate fungi remains of the Kalimotallus pertussus type, a new combination Neomycoleptodiscus pertussus was made. And this is example from India. This is near modern from Holocene, from Ashmuti and Paravur wetlands. This near modern, as an example that this form was not only in the past, beginning from the late Cretaceous, but it is also present near modern deposits of India. It is, of course, found in many localities from India, fossil localities. And these are modern relatives, Neomecoleptodiscus venezuelense and Meocoleptodiscus disciformis. As you can see here, they have porate cells. This in Neomecoleptodiscus venezuelense, and in Mecoleptodiscus disciformis, these are characteristic pores can be visible. Sorry. Contrary to Calimotalus pertussus, another fossil Calimotaloid species, Calimotalus quilonensis, has pores found only in outermost rows of cells of the Sporodochium, slightly elevated walls forming collarette around pores. These features suggest taxonomic affinity with some species of the modern genus Myocopron. For this fungi remains a new fossil genus, Myocopromyces, was created with fossil species Myocopromyces quilonensis. And this is Indian forms from Miocene Kerala, formerly Calimotalus quilonensis, and now Myocopromyces quilonensis, also a family Myocopronaceae. And modern relatives, Mecoleptodiscus brasiliensis. As you can see, this in outer part of cells, the spores, and this pores with collarette clearly visible. This is a modern form from Mexico. And another, formerly Mecoleptodiscus freycinitiae, and now it is Myocopron freyciniticola, clearly visible. These pores also here visible, and here with collarette also. This is a very similar form to this Mecoleptodiscus brasiliensis. And last one, Calimotaloid fungi belonging to the fossil genus Cribrites differ from both Neomecoleptodiscus and Myocopromyces quilonensis in the size and the orientation of the pores and in the arrangement of the cells of the Sporodochia. No modern related to this genus were found. And this is an example of Cribrites aurea from Australia, described by Lange, and this is from India. Ratnangitherites hexagonalis formerly, and now we consider them as a Cribrites hexagonalis. This form is from Miocene, Ratrangibets, Mashaharata, India. And this can clearly, this difference that our pores are very large, covering near the whole surface of the cell of this Sporodochia. And the fossil Calimotaloid fungi from India was reported also from Sivalik deposits. After Saxena and Tripathi, this was two forms, Calimotalus assamicus and Calimotalus raimanayumani. And these forms also should be considered as Neomecoleptodiscus. These Calimotaloid fungi are important as paleoecological proxy. Modern representatives of Myocopronaceae family related to the fossil Neomecoleptodiscus pertussum and Myocopromyces quilonensis have a geographical range limited to the tropical and subtropical areas of the globe. Those Neomecoleptodiscus and Myocopromyces quilonensis should be considered as a reliable climatic proxies of the warm climate conditions in the past. Like most of their living relatives, they were probably saprophytes on decaying leaves. So this fungi has important, very important for paleoecology. And these are references used here. Thanks for your attention.
We have time for one or two quick questions, if you have. Angela, I can see you after a long gap. I am finding you. How are you?
Yes, I'm fine, thank you. Yes,
can you remember?
Of course. Yes.
So Angela has certain question. Please go ahead, Angela.
Yes. Thank you very much. I'm enjoying being here. My question to Gregor is concerning, I don't remember the name, the first group you presented from the Holocene of India. You said that the modern relatives are living in South America.
Yeah.
There are no modern relatives in India or in Southeast Asia that could be related to that? And if so, why? What do you think? Why is it?
Sorry, thanks, Angela. I would like to explain this problem. These are, is it visible now or not?
No.
Okay, I don't know why it is not.
There is your PowerPoint program.
Yes, PowerPoint. Yes, I would like 5. Okay.
Yeah, maybe you just in the sake of time, just answer.
So I understand. These modern relatives were found in South America. However, Lange described from northern Australia not described forms identical to Calimotallus. And as you can see in my presentation, in India, especially in India, in Holocene deposit was several papers concerning Holocene deposits with preserved remains of Calimotallus-like fungi. So it surely exists in India, surely exists in northern Australia, and probably in most part of the world. However, there are no mycologists that are working with this fungi, and the only published allowed paper to me was this from South America concerning Mycoleptodiscus disciformis, and Neomycoleptodiscus venezuelense was published 2 years ago, so it is quite recently published.
Okay.
Okay. Surely it occurs in all of the world, in my opinion.
So it's still, you think it's still in India today?
I'm
probably.
Sure.
Okay,
it's very
thank you.
It's very probable.
Thank you.
Thank you again, Varavik, for your nice presentation. So it's time to shift for the next presentation, that is by Dr. Anusuya Bhandari. She is from Birbalsan Institute itself, and she will present her work on remarkable Miocene vertebrate fossils from Kutch, Western India. Dr. Anusuya Bhandari, please.
Yes, sir. So can you see my presentation slides?
You are presenting now, it will take time.
Okay.
Not yet.
I have already shared my
No, it is not. It has not been shared.
शेयर हो गया डाउन करके पीपीटी शुरू कर दे डाउन डाउन करो डाउन राइट टॉप राइट स्टार्ट योर पीपीटी
अरे स्टॉप शेयरिंग हो जाएगा.
just
Again, you keep your PPT file open, and then come to your left side icon, that is Google. Yes, it's coming now.
Yes.
Okay.
Now full screen.
Full screen.
No.
Full screen.
Just a minute. Full screen.
No. Open your PPT.
Yeah.
Full screen.
Microsoft Edge क्रॉस करो, लेफ्ट राइट हैंड साइड में उसको क्रॉस कर दो पहले.
इसको क्रॉस करो.
Then there are some buttons, hide and stop sharing.
No. Come to the bottom.
Bottom.
एकदम बिल्कुल नीचे. हाँ. Right. Now it's coming.
Okay.
हाइट पे भी लगा सकती है वो, उसको चला.
हाइट को कर सकते हैं. Go to your first slide.
Yeah. Just a minute.
That is all.
Full screen.
Full screen. Oh, Microsoft Edge is
There is some problem.
that's okay. That's okay. It's okay now.
I'm Dr. Anusuya Bhandari, working on myosin traditional biota of non-marine packages of Kutch, Western India. And as you know very well, that earliest neogen mammals are poorly recorded from central Kutch, while the diversified mammals are recorded from the northwest Himalaya, Shivalik area. But it is less known and it is rare from the out of Shivalik. So here I'm going to show you the remarkable Miocene vertebrate. Can you close it? Vertebrate fossils from Kutch, Western India. So Miocene is very important time slice because it is very interesting for a paleontologist because major upliftment has taken place during that time period and it affected the climate driven installations of monsoon systems, which changing the terrestrial biota and resulting the migration of many mammalian clade during that time period. So it marks a bio event and it was the witness of rapid development of the vertebrate, and this time period is very important because appearance of many hominoids from Africa into India. So this is the geological map and it is showing the fossils locality. Here you can see I'm working here in Tapar and Pasuda area, and we did extensive field work in Pasuda Tapar area and found the late Miocene sections and locality in central Kutch area. So this is the part of central Kutch, and here you can see this is the yielding horizon where we got fossils from the ferruginous conglomerates. So these are the neogen sediments, and a paleontological study carried out here to know the biostratigraphy of the regions. So in initial state, what we thought that it is early Miocene in age. However, the recovery of Hipparion and the other mammals suggested that early late Miocene packages, and we have given age on the basis of age diagnostic component. So here you can see this is the three-toed Hipparion tooth, and which is very significant, and presence of this Hipparion in our collection, it indicates that the late Miocene sediments in the regions, it means the younger horizons are present in the regions. So Hipparions are basically tridactylite, three-toed equids that profusely found in the middle Shivalik, and you can distinguish it very well on the basis of protocon that is present in the upper molar, and they have isolated protocon. Here you can see, and this Hipparion is evolved from the Meteriac, and it evolved in the North America, and then they migrated towards Europe and Asia through the land bridge connection. So it is very significant for a biostratigraphy point of view, and it marks a bio event, that is a Hipparion datum event around 11.8 to 11.5 to 10.8. Beside that, we got the age diagnostic mammal, that is Kutchacorus salinus. Kutchacorus means Kutch ka pig, and the other mammals is Twitter conodont indicus. You can see the ferruginous conglomerate where we have recovered these weeds and Hipparions. So this is the marker. So now I'm going to show you a very significant fossils, and that is a remarkable fossils, and we can say it is the first and oldest record from Kutch area. Previously, it was not recorded from Kutch area, and this paper is published in PLOS 1. So this is the outstanding output and landmark discovery from Kutch area, and during field trip, this hominid maxilla we found from Tapar locality. And this is a discovery. So this new discovery will help to understanding the evolutions of great ape. Shivapithecus means ape from the Shivalik hills, Shivalik ka Vanar, and that having a low and bunodont teeth, and the enamel is always thick. So it upper and lower molar have lex singular. So we can distinguish it with the other genus. So Shivapithecus is known from Shivalik hills of Pakistan and India, also from the Potwar plateau in Pakistan, Ramnagar, Harital Younger, in the northwest Himalaya, Churia Hill in Nepal, and now it is recorded from Kutch. So the discovery of this Shiva Pithecus from Kutch, so it's constitute the first locality, very first locality in the Indian Peninsula that is the part of out of Shivalik. That is not part of Shivalik, that is out of Shivalik. So it is very significant in the geological time of scale because it added one more area, one more locality, and it filled a time windows of approximately 11 millions in the evolutionary history of hominoids remains in the India. Now I'm going to show you the another mammals, that is a small terrestrial mammal, Rodentia, in our collection, which is also very significant for a biostratigraphy point of view, and it is used as a biostratigraphy tool for dating and correlations because rodents are the best age diagnostic components of micromammalians. And after macerating 1500 kg samples in the lab, BSIP lab, we have recovered diverse mammal assemblage in our collection. It consists of squirates, rhizomid, chrysitid, and murids. So murids, here you can see murid is the most dominant group in the Cenozoic sessions in the late Miocene time periods, and Progenomus morgane, which is index fossils, and we can say it has a short range species, and it is very primitive, first time recorded from Kutch area. And while the advanced form is recovered from the near Harital Yangal Lathiani from the northwest Himalaya. So it indicates early late Miocene age. So this study highlights that Wiesen had murines, primitive bamboo rats, chipmunks, and the other rodents in our collections. So beside that, we got a diverse assemblage of mammals in our collections. So we have here you can see this is the carnivore, and then Serenians, then bovids, then we have giraffids, honcore. We have fish also, prestige sawfish, and rhinos in our collection. Then Dorcatherium minus, that is triangulates. And we have, rather than this, we have scutes of turtle here you can see, and scutes of crocodile and crocodile tooth. Plus we have Dinotherium syndiensis molar, that is broken, slightly broken, and it is smaller than the Endicus. And Endicus is recovered by Singh et al. Recently. So we have two bats in our collection and one insectivores, which is very significant in the context of paleoecology point of view. Then we have unworn teeth of cyprinid fish, and cyprinids are the freshwater fishes. They have pharyngeal teeth in the fifth gill arc. They have throat teeth. So they are very significant for our paleo environmental studies. They are typically freshwater. So now I'm going to give you update checklist of neogene mammals, because aim of this study is to provide the update checklist of neogene mammals. So here you can see in my collection we have Rodentia, we have bats, we have carnivores, we have hominids, we have Serenians, we have equids, we have Proboscidea, rhinos, anthracotheres, sweets, sanithares, triglyphids, giraffid, bovid, a very diverse assemblage. And beside that, we have pisces also and reptiles in our collection. So I'm going to conclude my slide, and at last I can say that the presence of Hipparion in our collection, which indicates that younger horizons in the regions, and it is not older than Hipparion datum event. So discovery of Shiva Pithecus from Kutch Basin represent a southern range extension of Miocene hominid in the Neogene time period in the Indian Peninsula. So it means it extended the biogeographic range from north to south by 1,000 kilometers from the previous finding. And then the assemblage of rodent is very significant in the context of paleobiogeography, because during that time period, homogeneous mammal in the Indian subcontinent, and it is very significant biostratigraphy point of view and support an age of early late Miocene, that is more than 10, means 10.8 to 10 million year ago. So based on rodent study, we have given early late Miocene age to the horizon. Now we have a diverse faunal assemblage. Previously I said that we have Proboscidea, equids, anthracotheres, sweets, number of mammals in our collection from the central Kutch. We suggested that the ecosystem was very special, well-established. Subtropical ecosystem was there during that time period, and moist environment was there. It means it was more slightly humid during that time period. Thank you so much.
Thank you, Anushya, for your presentation. A very good presentation, Anushya. And I think there is time for one or two quick questions. If you have any questions, please. Is there any question? Yes, I can see Rachna Subba. She has a question: does the new found assemblage of mammalian fossils throw any light on the proliferation of mammals in this region during the Miocene? Anasuya, can you hear?
Yes, sir. Can you repeat the question? Is it in chat box? Is it in chat box?
Yes.
So I can give answer. I can give an answer over there.
Yeah, please answer.
So I cannot see the chat box.
I'm repeating the question. Does the new
found assemblage of the mammalian fossils throw light on the prolifications of mammals in the region during Miocene?
Yes. This is the question. Dr. Bhandari, please respond.
Yeah. I'm just.
We are running out of time.
Which mammals? Because every mammals have their own, significance during Miocene time period.
Okay. But
so she's asking about which assemblage?
Proliferation of mammals during the Miocene.
Hello?
Yes, we are hearing. Your mic is now off. Turn on, please.
Yes, sir. The screen is problematic, actually.
Any explanation from you?
So, actually,
may I answer? May I answer? Actually,
Dr. P.P. Mishra, please respond.
With the withdrawal of the sea and with the beginning of the terrestrial conditions in this coastal Kutch region, with the beginning of the land conditions, we start getting several now with initially we used to get only the proboscidean remains, but now with Dr. Bhandari's report, we find that large and varied terrestrial fauna, mammalian fauna specifically, we do get in Kutch region also. So that is very, very interesting. Definitely, apart from the Shivaliks which we have in the northern Himalayan part, we had in this part also with the say in the middle Miocene or so, we started having so much of varied mammalian fauna. That is my submission. Dr. Bhandari may.
I can give answer. Actually, there are many faunal assemblage in my collection, so I was answering of her questions because it's so light. Previously, we are getting the early Miocene mammals, that is, proboscidean, and which shows the connections. And after that, there is a middle Miocene packages are there, and after that we are getting a fossils assemblage, and you know, this assemblage is very important in the late Miocene time periods because it shows the mammal evolutions during that time period, and we are getting the variety of fauna. Not only, I mean, not only Proboscidea, but hippos also, hipparians also in our collection, different varieties of fauna. It means you know. Condition was subtropical, dense forest was there during that time period, and after withdrawal of sea we are getting these packages.
So I think Rachna got her answer. Now I think Dr. Mahesh Thakkar has some question. Dr. Mahesh Thakkar, you raised your hand. Yes. No? So if there is no more question, we are moving to the next presentation. That is by Abhishek Pratap Singh.
Yes, sir. Yes.
And the presentation is on new micro-mammals from the middle Miocene of Ramnagar, Udhampur district, Jammu and Kashmir. And the abstract has been authored by Abhishek Pratap Singh, Ramesh Kumar Sehgal, and N.P. Singh.
Yes, sir. I'm Seri.
Yeah, please go ahead. Without wasting any time, we are
yes, sir,
actually,
yes, sir.
Yeah, you please start your presentation.
And so, ma'am, please.
Yeah.
Your entire screen is showing.
That's Anusuya's screen. Anusuya is presenting that.
Sir, okay now?
Abhishek Pratap Singh,
Yes, sir. I'm sorry.
please start your presentation.
अरे यार, इधर से। Yes, sir. Slide is showing, sir. Hello?
Yes.
Yes. Good afternoon, everyone. My name is Abhishek Pratap Singh from Wadia Institute of Himalayan Geology. My presentation topic is
I'm sorry, please, I'm interrupting you. Please have your full screen.
It is in full screen, sir.
No. You come to the next icon on the left. That is a bookmark there. No. Where you are, just yes. Oh. Next, left. Oh. This one. Yeah.
Okay. Yeah. My presentation topic is my new micro mammal from Middle Miocene of Ramnagar, Udhampur district.
You are not there actually. You go to the next left. There is a book-like thing. Read. The icon will say readable.
It's not full screen yet, Abhishek.
Abhishek, please left one.
Oh.
No.
F5.
No. Go to right. Again right.
F5.
No. Left. Oof.
F5. Right. Shortly.
This one is slide show.
No. Next one. Next left.
Reading.
Yes.
Reading. Okay.
Yes. It's okay now.
Okay. Sorry, sir. This is geological map of tertiary bed of Northwest Himalaya modified after Rangarao. In it, I mainly delineate lower, middle, and upper Sivali with different colors of green, orange, and yellow. And the edge of Sivali is bounded from south SFT and from north MBT. As Pilgrim divided Sivalik into three subgroups like lower, middle, and upper Sivalik. Lower Sivalik is further divided in formation level like Kamliya Chinji in lower Sivalik and Nagri Dhopatan in middle Sivalik as Tetrod, Pinjar and Boulder in upper Sivalik. and the age range of Shivalik is 18.3 to 0.22 millennium on the basis of magnetostratigraphy of Shivalik area. This is the geology of Ramlanga. Figure B in this slide showing geological map of Jammu area and of Shivaliks, and figure C is showing a different fossil locality of Ramnagar from where earlier worker recovered mega and as well as micro mammals. And this litholog is prepared from Dihari locality, and in this locality lithologically vary from mudstone, sandstone and interformational conglomerate. In respect of Shivaliks, interformational conglomerate bed is more important for as concern of micro mammals, and we collected sample of that red spot of litholog of pseudo conglomerate. Figure D, photo D is field photograph of Bihari locality. The methodology that we use to recover micro mammal from pseudo conglomerate layer is oil water immersion technique. In this technique, we first dry the sample and broken sample into small pieces, and broken pieces soak in kerosene oil for 7 to 8 hours. After that, kerosene oil poured out, and entire sample keep in 7 to 8 hours in water. After this process, hard material is get fragmentary, and this fragmentary sediment is passed through wet sieving with different size of sieves. And in this process of sieving process, after sieving process, we remaining material is dried, and micro mammal from this material is picked under the binocular microscope. During this process, we got some new specimen of Tupaya genus and Galerix, as well as some rodent that already reported by earlier workers. This is a insectivore that we got in Bihari locality. This is the first finding in Indian Shivalik of middle Miocene. These are as we study this sample on the basis of size and shape analysis, with as earlier reported Galerix species in Potwar Plateau of Pakistan, we can say this species fall at Galerix vesselsi. As a Galerix species, two species of Galerix found in Potwar Plateau of Pakistan, like Galerix vesselsi and Galerix rutlandi. Galerix vesselsi is as older, its time range 19 million to up to 14.1 billion, and Galerix rutlandi is from 14 to 10.2 million. As a statistics measurement of this specimen, we analyze in past software. This specimen fall at near to Galerix vesselsi species. So this species we can correlate at Galerix CF vesselsi. This is Trisu species that we reported from Dihari locality, and for morphometric analysis of Trisu, we use CT scan photograph in Amira software. In this Amira software, we digitize landmark. Approximately 13 to 40 landmark is required for morphometric analysis, and each landmark have their own coordinate, and this coordinate we transfer in past software. In past software, we perform this comparison with living and extinct Tupaya species. During this in this graph, our species fall in Tupaya species, but only single specimen we cannot keep it in Tupaya species. So at family level, we identify it as Tupidae family. Keep it in Tupidae family. This is CT scan photograph of Tupaya, as well as some additional rodent species that we identify in Bihari locality. As a Tupaya, this Tupaya species specimen is now oldest one. As earlier, Tupaya species in Pakistan reported from 10 million year, and in India, Tupaya is reported from Tetrod formation by Datta et al. 1975. From Tetrod formation, and Vasista et al. Is in 1979 found in Haritlanger which is in Middle Siwalik. Now this finding is from Middle Siwalik, up from Middle Miocene and up Lower Siwalik in India. Additional to Tupaya, we also reported different rodent species from Bihari locality like Chimus indicus and Chimus sivilensis and some marine species, family species. We compare all these Tupaya species and rodent species in past observed on the basis of their shape and size. We compare these Tupaya species already reported from Siwalik localities. Our species fall some other from some deviate from Tupaya species, but we require more species specimen to give it as a new species. Like that, we compare rodent also in parts observed to know the specimen called what type of species, and we compare shape and size of the recovered fossil specimen to already reported one, and this specimen of Chimus indicus is fall in that region which is also equivalent to Chimus indicus, so we give its name as that one. Like that, we compare Chimus sivilensis also, and after other specimen we also compare on the basis of shape, size with different rodent species reported from Siwalik and other Siwalik and other localities. The Tupaya specimen described here from Ramnagar dating to Middle Miocene is significant extend their temporal range in Tupaidy evolution. And we see few possibility further specimen confirm the presence of Gallic specialty in the Bihari locality. Like the age range of Gallic specialty is from 19 to 40.1 billion in Pakistan, but in India, that locality we got this specimen, that age is 13.5 to 13.6 to 13.2 on the basis of rodent biochronology. So on the basis of that, we can, if it possible to give this specimen name Gallic facility, then last appearance data are the Gallic facility increase up to 13 point or 12.5 approx. And second possibility is that the Bihari locality age extend back in geological age to equivalent to as a base of Chinji formation on the basis of this Gallic facility. And the Bihari locality specimen represent previous undocumented specimen that is distinct from the Gallic facility and Gallic rutlandii on the basis of their size and shape. We can give new species further specimen required for that region. Thank you.
Thank you, Abhishek Pratap Singh, for your excellent slides and excellent presentation, as well as well done figures by CT scan, etc. And we are running out of time, but if you have any one question, I can allow right now. Any question from anybody? Is there any question in the chat box? No question? Anybody? Dr. VP Mishra, please, if you have any question. Dr. VP Mishra, if you have any question.
No questions. Good presentation.
Okay.
Good presentation.
Right. So if there is no question, we are moving towards the next presentation, that is by Adrisuta Ghosh, and she is from Indian Statistical Institute, and she will present her work on documentation of change in diversity pattern of Kutch gastropods across the Paleogene-Neogene boundary. So you please start your presentation, Adrisuta.
Okay, sir. Is it visible, sir?
Now go to full screen. Yes. Yes, perfect.
Okay.
You can hide this one, the
Okay.
lower one.
Sure, sir.
Yeah.
So the title of my presentation today is Documentation of Change in Diversity Patterns of Kutch gastropods across the Paleogene-Neogene boundary. And the objective of this study is to demonstrate, as the title suggests, to demonstrate the changes in diversity pattern of Kutch gastropods based on three aspects. Now, diversity is a broad term. We have considered only three aspects of diversity for this study: the taxonomic aspect, the morphological aspect, and the ecological aspect. Now, for the Cenozoic stratigraphy of Kutch, our study is based on the Kutch Basin, which is present in the western state of Gujarat. And it is a pericratonic basin which records depositional succession from Triassic to recent. And our study is concentrated on the Cenozoic sediments here. About here, our locations are all around this place. And a pictorial representation of the place is given here. As you can see that the core is occupied by the Deccan traps, and the Cenozoic sediments, they wrap around the Deccan traps, and as we move westwards, we encounter the Paleogene-Neogene boundary, that is Younging occurs in this direction. And now for the tertiary stratigraphy, you can see that the Deccan trap occupies the oldest position, and that is overlain by the Mathanamud formation of the early Paleocene. Mathanamud is practically unfossiliferous, containing some floral assemblages, which is topped off by Naredi, Harudi, and Fulra formations of the Eocene. Now, Fulra limestone and Naredi formation are also comparatively sparsely fossiliferous, whereas Harudi is very richly fossiliferous. The Oligocene formation of this region is the Maniara Fort formation, and the Miocene formations are Chasra and Karinadi. Now, early Miocene formations from where our specimens have been collected. Now, for the regional geology and depositional history of Kutch Basin, as we know that the Kutch Basin opened as a result of the breakup of Gondwana, and in the north, there was the Nagarparkar Hill, and towards the south, there was the Saurashtra Peninsula. When the middle portion experienced some rift, the Kutch Basin got created, and this basin basically formed a westerly dipping epicontinental sea, in which deposition took place later due to several transgressive events. And then siliciclastic sedimentation took place, which was cut off by the Deccan Trap volcanic activity. And the Cenozoic sedimentation began at Kutch in a stable shelf environment. And now the materials and methods of this study. Now, for the taxonomic study, the data has been collected from the published literatures and also our institute's own collection. And all the specimens are collected from the 6 lithostratigraphic formations, as I had mentioned earlier: the Naredi, Harudi, and Fulra formation of the Eocene, the Maniara Fort formation of the Oligocene, and the Karinadi and Chasra formation of the Miocene. Now, we have made a species list that has been prepared with the temporal range, which I'll be showing shortly, from Eocene to Miocene. Now, this data comprises of 304 species belonging to 128 genus and 33 families. However, for the statistical analysis, we have only chosen the families whose species count was more than 3. Now, for the morphological analysis, we have selected the body forms, the common body forms that are seen in gastropods, and we have divided our species into them, and we have evaluated the statistical analysis using pie charts. And for the ecological diversity, we have used information that we obtained from the paleobiological database, and we have included certain life habit and diet types such as epifaunal carnivores, epifaunal grazers, herbivore, omnivore grazers, suspension feeders, infaunal carnivores, and semi-infaunal suspension feeders for our ecological statistical analysis. Now, this is the species list that I mentioned. So as you can see, we have plotted the species and the and the black line denotes the...
Unfortunately, Adrushita, we cannot see. This font is not visible. Anyway, continue. Please continue.
Okay, sure. It is present in the abstract, this chart. Okay.
Okay, no issue. You please continue.
So the results are that we can see that 17 families are reported from Eocene and Oligocene and 26 from Miocene. So there are a total of 27 families, and 26 of them, with the exception of family Pleurotomeridae, all families are reported from Miocene. And we also see that 4 new families appeared during Miocene: the Cyametidae, the Melongenidae, the Terebridae, and the Turidae family are the new families that appear in Miocene. Now, for the morphological diversity, as you can see, we have prepared pie charts showing the species diversity, the species abundance. And we can see that for Eocene and Oligocene, the opconic shell form is the most abundant one. However, when we move to the Miocene, the fusiform shell forms became the more dominant shell form, though the opconic remains second, but still the fusiforms dominate. And another more important thing that we noticed during the Miocene is that we can see that there are only 6 to 7 types of shell forms present in the Paleogene, but in Neogene, we can see that 11 kinds of shell forms were reported. So there was a huge increase in morphological diversity as we entered Neogene from Paleogene. Now, moving on to these are the results which I just summarized. So moving on to the ecological diversity. Again, we have taken the groups, the life habits and the diet groups, and what we have done is we have counted species for each type. And as we can see that during the Miocene, this count of species increased quite a lot from that of Oligocene and Eocene. Now, for the results of the ecological diversity, we can see that the epifaunal carnivores, the epifaunal grazers, the epifaunal omnivore grazers, semi-infaunal suspension feeders, all of them have increased during the Miocene. And there is like the species count increased. And there is another trend which is noticeable, that is during the Oligocene, in the 3 most dominant ecological types, we can see that the number of species dipped during the Oligocene, and then it picked up and reached its highest point during the Miocene. For these 3 cases, we can see that. And these 3 are the most dominant ecological forms that are present there. Now, so we can say that there was some sort of issue that was going on during the Oligocene, which can either be biased, that like not many species were collected from Oligocene, or it could be that there was some sort of limiting factors that controlled the number of species. So that's, and for conclusion, we can say from the taxonomic study, we saw that new families are being introduced, and almost all the families that were already present continued. So we can say that the taxonomic diversity increased a lot. And for morphological diversity, again, we saw that new shell forms got introduced, and the previous ones were already continuing. And for Neogene, even though there was no new habits or diet forms as such, but the species count or the species abundance in each of the groups increased in most of the types. So those are the 3 concluding remarks and references and acknowledgements. And thank you for letting me present in this platform today.
Thank you very much, Adrisuta, for the nice presentation. And it's now open for discussion.
Yes, sir.
Anybody else? Dr. Mahesh Thakkar, we want to hear from you. Yes, Angela. Angela, you have some question, I think.
Thank you.
Yes, please.
Thank you. Yes, well, I have no idea about gastropods, but I would like to ask, may it be that there is a connection between morphological diversity and ecological diversity? This goes in
yes,
parallel?
that is a thing, that some shell forms and some morphologies are supported in specific substrates, and that is the case for the ecological diversity too, that some life habits and some life habits are more supported than the other. We are still looking into it. There is no conclusive result for that, but we will be looking into it.
Dr. Mahesh Thakkar? Yes, please go ahead. Dr. Thakkar, you raised your hand. Is there any question? Please turn on your mic.
Unmute.
Please unmute. Torsten, you have some question, I think. Yes. Yes, go ahead. Yeah.
Go ahead. Yeah.
Good afternoon. Good afternoon.
Is it possible that there are climatic implications of this diversity change you observe, connected to the plate tectonic movement, for instance?
So yes, I think there is such a possibility, but right now, as it's a preliminary study and we have just dealt with the data, we have not gone into that implication, so we will be doing that. So.
All right, thanks. But very nice story.
Thank you, sir.
Thank you.
Adrishita, may I ask some question
yes,
to you?
Yes, sir.
My first question is this, that do you think that the lithological features of the Oligocene, specifically you are talking about the Oligocene and the type sections are your Chattian type section, that Bedmoti section or Bednani section, these are the two sections in Kutch. Okay, do you think that lithology may be responsible for the yield or for the productivity or for the, in fact, your recovery of fossils, gastropod from those, in fact, exposures?
So I have not actually.
Because the Chattian, you have not in the field. You have been in the field.
I have not visited the field yet. I have just dealt with the data. So.
Okay.
So you are talking about the preservation quality.
Yes.
Okay.
Because you know that in case of fossils, we are mainly concerned about the preservational factors, right? In fact, if you go to the Bedmoti section or Bednani section, those are the Chattian sections of the Oligocene. If you go there, the limestones are so hard and full of fossils, but it is very difficult to recover or extract the gastropods or any fossils from those.
Okay.
Only thing is this, you have to do the thin sections. No other possibility of doing of studying those fossils. Okay. You can talk to your boss or talk to your seniors regarding this. And my second question, not a question rather, just few days back I saw it. Till date we are following the sequence stratigraphic framework of Biswas 1992 that was published in your Journal Paleontological Society of India in 1992. But you please consult a recent paper, and I also came across this paper through one of my students actually, and that is a very good paper, and they have actually compiled the offshore and onshore sequence of Kutch, and that is by Octavian and Alok Dabe, 2017, right?
Yes,
that was published in Journal of Petroleum Geology. Please go through that paper. And my last submission is that please check the spelling of Kutch. Okay? Everybody, even Anusuya Vandari also, I have not told her because she is from our institute only. When you are giving any formation name or any sort of name, you please write KUTCH, no issue. But in fact in the government site, you will find the spelling of Kutch is entirely different. It is KACHHCH. Okay? You please follow this when you are writing your paper. For Kutch district, you please write Kutch like this.
Yes,
Okay,
so I've
you can
seen
consult with, yeah.
in the
Okay,
catalog paper.
yeah, please, my colleague Vivesh is also adding something.
Yeah, this is not the first time, but quite numerous times I've come across the spelling of Kutch, like K-U-T-C-H, Kutch, is the formal name for the basin, if I'm correct.
Yes.
So, but K-A-C-H-C-H-H is the formal name for the district. So whenever you're talking about the basin, write K-U-T-C-H, and whenever you're talking about the district or the region, then you have K-A-C-H-C-H-H.
Same thing happens to Naradi also. You see,
And
Naradi is a formation. You keep Naradi as such. The village name is Nareda.
Yeah.
Okay.
Sir, yes.
Okay. So somebody writes Nareda formation, but Nareda formation is not right. Naradi formation is the right, and Nareda is a village name, after which the name Naradi formation has been given. Okay. So thank you again very much for your nice presentation. Coming to the next one, that is fossil wood of Cordia from Tipam sandstone formation of Northeast India, and this will be presented by Harshita Bhatia. The paper is co-authored by Harshita, Dr. Gaurav Srivastava, and Dr. R.C. Mehrotra.
So, hello everyone. I am Harshita Bhatia from Vivekananda Institute of Paleosciences, and I will be presenting here the fossil wood of Cordia from the Tipam sandstone formation of Northeast India. Fossil woods, they are important in building the paleoforestic and in reconstructing the paleoclimate of the area. The present fossil wood was collected from the Difu, Assam, Northeast India, and it is from the late Miocene. Here, these are the fossil woods which were collected from the locality. Based on the lithostratigraphy, the Tipam sandstone formation is considered to be the late Miocene in age, and here this is a systematic description of the fossil wood. It belongs to the Boragini order and is placed in the form genus Cordioxylon indicum, species Nova. Here comes the description part. So here this is the transverse section, which is showing the arrangement and the distribution of the vessels, parenchyma, rays, and other features. This is the enlarged DS. Here we can easily see the distribution of the parenchyma. These are the basocentric to confront exit parenchyma. Here these are the banded parenchyma. These are the rays. Then here these are the fibers. This we can see here, these are the vessels which are filled here with the tylosis. Here is the transition longitudinal section or the TLS, which shows here the rays and also the prismatic crystals present in the parenchyma. This is the enlarged TLS, which shows the detail of the ray cells. Here we can see the vessel ray elements, which are having a simple perforation plate, and also the pits which are present here. The radial longitudinal section of the RLS is weakly heterogeneous and is mainly composed of the procumbent cells and 1 to 2 cells of the upright of the square cells. These are the intervessel pits which are alternate and bordered in nature. RLS here shows the vessel ray pits which are also similar like the intervessel pits, and they are also alternate and bordered in the nature. Here we can see a clear picture of the vessels surrounded by the parenchyma, and these are the rays. Here these are the prismatic crystals which are present in the ray tissue cells. Here we can also see a very clear picture of the crystals which are present in the axial parenchyma, and also a somewhat storied parenchyma present here. So this is the map showing the distribution and the fossil records of the Cordia. We can see that the Cordia is distributed worldwide, mainly in the tropical to the subtropical areas, but the fossil record of the Cordia, they are extremely limited or poor. They are mainly known from the late Paleocene of Argentina, Paleocene of Egypt, Miocene of South India, and from the present fossil locality. So the previous fossil records from the locality, it includes the genus, Cenozoic, Glutazoilon, Liaonozoilon, Cassinium, Cynometrozoilon, Terminatozoilon, and Paraphyllantozoilon, indicating an evergreen to the deciduous nature of the textiles. So, based on the occurrence of the cordia in the northeast India in association with the other evergreen to deciduous textiles such as the Canarium, Gluta, Linea, Cassia, Cynometra, Terminalia, and Phyllanthus, it indicates that the presence of a tropical evergreen to deciduous forest was surviving there at that time during the Miocene. It can also be concluded paleoforestically that a tropical warm and a humid climate was present there during the depositional period. So, a very thank you for listening to me. Thank you.
So her presentation is over. Any question?
Dr. Spicer has
Yes.
Okay.
Thank you, Harshita, for the nice presentation. And I think Bob has certain questions. Bob, you can ask.
Yeah, I gather from looking at the abstract and from what you've presented that the age of the Tipam sandstone is late Miocene. But can you tell me on what basis it is assigned a late Miocene age?
Based on the lithostratigraphy, it is assigned a late Miocene age. Based on the work of the Karnataka.
Okay, thank you.
Thank you.
Any other question? Any question? So if there is no question, we are moving to the next presentation by Hung Ba Nguyen, and it is from Chinese Academy of Sciences. Hung, you can present your paper, first fossil pod record of Mucuna, that is from Papilionaceae fabaceae, from the Miocene of Vien Bai Basin, North Vietnam. Hung, you can present your paper.
Yes.
Please go ahead.
Sorry, how can I say my screen?
It is in the PowerPoint?
Yeah.
Okay. You keep your PowerPoint open.
Yes.
And now come to the present. Now, yes, you are presenting now.
Yes, okay now.
Now come to the full screen. You come down, please.
Yes, okay now.
Next one, next left. Ready? No. Left, yes, this one. Oh. Previous one. Next left.
That's left.
There is a bookmark there. No. Right, please. Next right. Yes. Oh, you have missed that. Next.
That's left.
Left. Just one. Yes, this one.
Reading view.
Reading view.
Yes, is it okay now?
It's okay.
Yes, yes, I'm sorry for the problem. Yeah.
No issue.
Yeah.
Please go ahead.
I'm going to talk about the research put for support record of Mukuna from Miocene in Basin, North Vietnam. And the genus Mucuna adan contain 105 living species and widely distributed in Pantropical area. 18 species in Mucuna show high morphological variation, especially in inflorescence, flower, fruit, and seeds. Based on geographical distribution and fruit character and molecular data, Mora et al. In 2016 divides this genus into 3 subgenera, including Mucuna, Seagrass, and Macrocappa. In contrast to another 2 groups, Mucuna Macrocappa include only 8 species and had much narrower distribution in southern China and northern part of Southeast Asia. And nowadays, Vietnam is known as a biodiversity hotspot in Southeast Asia. There are about 12,000 living species plant living species in Vietnam now. However, our understanding of plant diversity history is far from enough. So consequently, any new fossil finding is an important contribution to our knowledge. In this study, we describe the newly discovered and well-preserved fossil from late Miocene up in Basin, North Vietnam. And we further discuss the origin and early evolution of the genus and the paleoclimate during the Miocene in North Vietnam. And the fossils in this study were collected from the sediment of in Basin from sediment of Cổ Phúc Formation in Basin, North Vietnam. And until now, the age of Cổ Phúc Formation is still under debate. In this study, we consider the age of Cổ Phúc Formation in late Miocene based on the plant and mollusk fossil, which is supported by the latest study of nitrate and radiometric dating of deposits along the Red River fault zone. And there is only one specimen with counterpart containing samples were collected and investigated in this study. They were photographed using a digital camera, examined from previous permitted later and from posterior herbaria. The distribution of eastern mucuna was determined from GIF, and climate data for modern distribution were obtained from one cloud. And here is a figure showing our fossil support. And we assign our fossil support to mucuna conformit, both Uriana structure based on morphological characteristics such as legume line oblong, slightly curved, and compressed. The both body in preserved part is 15.4 to 35 centimeter long and 4.9 to 5.3 centimeter wide. And the apex is oblique, tapered, or shortly accumulated with a cone-shaped beak. And the port margin of a fossil is markedly constricted between seeds with a pair of smooth thickened marginal wings. And the seed chamber elliptic or ovate to nearly rectangular or square, and no less than 3 to 5 in number. And we're going to the next part is a morphological comparison. Our fossil supports so morphological character, same morphological characteristics with 8 genera in Fabaceae. However, the most important characteristics of fossil, such as the shape and size and both the and seed number, and the notable apparent of a pair of winged margins along the shoulder occur in present in Mucuna, which similar to our support, and our support should belong to the subgenus Macrocappa because they same morphological in morphology, and their species and their species in subgenus in this subgenus. And among these species, our fossil is different from all the species, except Mucuna birdwood in Anna, which similar to our fossil spot also has a pair of wings along the shoulder. However, given the current reservation, it is difficult to assign alpha support to a new PC. That's why we assign alpha fossil to Mucuna conformit but Urinana Kutch. And here is a figure. So there's a global distribution of living Mucuna. And as we know, Mucuna is a pan-tropical genus, and today, however, the origin and early evolution of this genus is far from enough, understood. They only questionable fossils were documented in South China. Third to our knowledge, our fossil support here is the first convincing macrofossil record of the genus, and it indicates and nowadays Mucuna still survive and widely distributed in Vietnam, and it indicates that Mucuna had existed in this region since the late Miocene. And Mora in 2016 suggested that Mucuna originated and then diversified in Asia during the Oligocene to early Miocene. Our new finding here supports this Asian origin hypothesis of Mucuna. And besides, Mora in 2016 suggests that Mucuna subgenus Dissolobim is the earliest diverging lineage within Mucuna. They hypothesize that the ancestor of Mucuna closely resemble to Mucuna subgenus Dissolobim, and the type both in Macrocappa and Mucuna is apomorphic character that appear during the intragenetic evolution of Mucuna. And our discovery here support their hypothesis that Mucuna had undergone a diversion from evolution in Asia by at least the late Miocene. And coming to the final part, this is a paleoclimate distribution implication. And our fossil support say morphologically similar to Mucuna but Uriana. They are liar and commonly found in dense or open woodland, rocky area, wet hillside, and valley. It indicates a similar habitat along the Red River Valley in the late Miocene. And here is a table showing the climatics that rank up the current fossil and Mucuna botulinana. And the figure indicates that the climate would become a bit more humid and getting to have a warmer winter and hot summer in nearby area since the late Miocene. And yeah, we also collected many other fossils from the same fossil site, such as legume, fern, and angiosperm. Among the fossils, Proteropermum only occur in tropical region. It's providing evidence that the vegetation of nearby area was tropical broadleaf forest in the late Miocene. That is similar to North Vietnam today. And it is that North Vietnam already had a humid tropical climate during the late Miocene. The floristic assemblage there had already been similar to that today. And this is all of my presentation. Thank you very much for watching and listening.
Thank you, Hung, for the presentation. It was a nice presentation. And there is time for one or two quick questions. If you have any question, please go ahead. Angela, yes.
Yes,
Angela,
me again.
Yeah,
thank you very much for this nice presentation. I liked it very much. My question is concerning the ecology of this liana. As you described it with humid valleys or something, isn't it rather a riparian element? Do you think it can this plant tell something about climate specifically? If it is growing in river valleys? Could you go to this second last slide where you wrote about it's growing in wet hillsides and valleys, rocky areas? Is it a climatic indicator, or is it rather
Yes, and it's the only showing indicate ecology in this area. And about the climate, we based on the other fossil also collected in this fossil site from the same fossil site is a Proteropermum. It's only occur in a tropical region.
Okay, so your climate reconstruction is based on all the taxa.
Yes.
Thank you. Everything. Okay, thank you.
Yes, thank you.
Now, I think Bob has a certain question.
Yeah, sorry, me again.
Yeah.
I was interested in the age. You showed a slide where you showed that there were some differences in the age call. Late Miocene based on plant and mollusks, and you make a reference to radiometric dating along the Red River Fault, but Oligocene based on palynology. We know that the Red River Fault began movement at the very latest Eocene and into the early Oligocene. And many basins along the Red River Fault were formed about that time. The redating of basins we've done in Yunnan seems to show this pattern. So I'm interested in where in the section the radiometric dating was done of this particular site, and whether or not these fossils could in fact be Oligocene rather than Miocene.
Yes. Thank you very much for the question. And we in this study we consider the earth formation is late Miocene based on the radiometric dating of hypothesis along the Red River. And Huang et al. In 2009, and they collected sediment sample in the same area in Bai province. Yên province along the Red River. Yeah, it's the
But
same
was it
formation
the same-
and.
Sorry, was it this same locality or one nearby? Because this is really quite
Yes,
critical.
it's nearby. Yes,
Okay.
it's
So it's
nearby. Yes,
not
and
this
you know, because it's yes, because I collect fossil here from the newly constructed road, and we have some interesting section for collecting fossil.
Okay. All I would say is if it is possible to revisit and find an ash within this section, it would be worth trying to date that, because I'm a little bit concerned given what we know about the different age calls in Yunnan and the timing of the Red River Fault activity. But yeah, very interesting talk. Thank you.
Yes, and thank you very much, Bob. And yes, thank you very much. Yes.
Thanks once again, Hong, and also to Bob Spicer. So to summarize this, in fact, session of NECLIME 2021, I have certain observations here. And first of all, I thank Professor Domning for the nice presentation of the keynote address on biotic evolution and biogeography during the Neogene in South Asia, based on Serenians and Seagrasses. And there are again 6 presentations by different authors on different aspects, that is ranges from fossil fungi from India by Vorobik, Anusuya Bhandari from the Miocene of Kutch. She actually presented some good fossils from the Miocene of Kutch. Then by Abhishek Pratap Singh from Wadia Institute of Himalayan Geology, he delivered a lecture on new micromammals from the middle Miocene of Ramnagar, Udhampur district, Jammu and Kashmir. And a very good presentation by Adrusita Ghosh and from ISI Kolkata, and she presented our paper on documentation of change in diversity pattern of Kutch gastropods across the Paleogene-Neogene boundary. Again, an excellent presentation by Harshita Bhatia on fossil woods of Cordia from the Tipam sandstone formation of Northeast India. And the last, but not the least, by Hong on first fossil pod or leguminous pod recorded from the Miocene of Vieng Bai Basin, North Vietnam. And these are all the presentation. I thank all the presenters and also to the participants, especially Angela, Dr. V.P. Mishra, and Bob Spicer for interaction and very, in fact, fruitful discussions. I think this was a very good session, and I thank you once again. And there is a break for tea break, or in US maybe a morning tea break, or in China it may be a cookie break, whatever it may be. In Germany it may be a lunch break. So that is break of different types. So please come again here at after 15 minutes, I think. After 15 minutes. 25. After 15 minutes we will resume. Okay. 25. I can see Professor Chamiel left. Okay.
It's only 10 minutes, right?
Yeah, 15 minutes.
Confused about the time.
Yes, me too. It's always a calculation, and we're going to start 5 minutes later.
Hi, Bob.
Hi. So I just tuned in to see you because I haven't seen you in a while.
Oh, that's so nice. Thank you. And I'm all nervous because I prepared a new talk and I had very little time to do it. So I was all.
Oh, good. We'll pounce on you when you get it wrong.
Yes.
Sure. Sure.
Yeah.
Anyway, you're doing okay?
Yeah, I was doing okay. I've been in Malaysia, but I just took on this new function, which is program director of Earth Sciences. And it's like I walked into a tornado. So,
you didn't volunteer, did you?
I did.
Ah!
It's my own fault. It's my own fault. I said, oh yeah, this is something new. I haven't done this before. And yeah,
Well,
Now
no
it's too
sympathy.
late
Yes.
to regret.
No sympathy
Well,
volunteered.
excuse me. I think we should continue with this afternoon session or evening session. Depends on where you are. And I hope the BSIP office will help me with technical issues, and I'm going to chair this part, which includes one keynote by Carina Horn and 6 short presentations. I hope we will stay more or less in time. And I'm glad to see Vandana Prasad again, and she is
Hi.
she's overseeing what we are doing, and probably will help me with issues. So it is my pleasure to announce the first presentation of this session, which is the keynote talk by Carina Horn on plant biogeography during the Eocene-Oligocene transition. And I'm looking forward to that.
Thank you very much. Let me first see if I can get it shown all right.
Yes, we had plenty of problems with that.
Is that so? Can you see my screen already?
It always takes a few minutes to load. We see your desktop.
Yeah, okay,
Yes.
so I'll put it on full then. Is this all right
Yes,
for you?
great. Yes,
Oh,
thank
that's
you.
pretty good, yeah.
Yeah.
Okay,
Okay.
well, yeah, thank you very much for having me at this session. And I realize that you've been all talking about the Neogene. And well, in my talk, I would like to take you a bit further back in time to the Eocene, and particularly to the Eocene-Oligocene transition, which I think it's fair to say in many ways, the history of the Eocene sets the scene for the paleogeography and so forth that we see in the Neogene. So I hope you'll find it informative. I would like to thank my collaborators, Washeng Wang, Giovanni Bogota Angel, Amber Woutersen, Natasha Barberini, Phil Jardin, Alexis Licht, Robert Morley, and of course Guillaume de Pont-Nivet, who have integrated a lot of our work in this one talk. So to start, I first would like to take you to this very familiar curve, a recently updated version of the temperature curve based on the delta 018, which is published in Miller et al. To show you the Eocene, which is a bit over 20 million years. And it's a very interesting time because, as you can see in the delta 18 curve and in the temperature curve, there is a lot of variation. So ranging from really high temperatures, greenhouse conditions in the early Eocene to cooling and dramatic cooling at the Eocene-Oligocene boundary. To highlight that temperatures in the early Eocene, according to Inglis, were about 10 to 16 degrees warmer than the pre-industrial time. So what is it about the Eocene-Oligocene transition that makes it so interesting? And so what exactly is it? So the Eocene-Oligocene is this period of cooling that in recent years, in the last, over a decade, has been identified as a period from now considered 790,000 years and starting at about 37.3 million years. And with the early Oligocene glacial maximum being the sort of shift to the greenhouse, from greenhouse to icehouse conditions. And there's some very nice reviews, an early one from 2007, Coxon and Pearson, but also more recently Hutchinson et al. Describe all there is to know about, well, maybe not everything, but most of what there is to know about the Eocene-Oligocene transition, how it has been dated, and the further background to it. And it suggested that the temperature drop at this boundary was about 2.3 degrees. And a lot has been said about what are the drivers for this change. And nowadays, it seems that CO2 drop is one of the main
drivers of temperature change, but also changes in global seaways and ice sheet growth on Antarctica were driving mechanisms. And finally, also orbital forcing is controlling the timing of the glaciation. Just about this changes in seaways, so it was particularly the opening of the gateways around Antarctica and the reorganization of the oceanic flow and the subsequent glaciation of Antarctica that is thought to have been a major paleogeographic, paleoclimatic event at the time. And here I show two different settings that are published in Hutchinson, which depend on which reference frame you use. And it's interesting to realize that the glaciation was only on Antarctica and that ice growth in the North Pole was much later. So we'll focus on this transition time, and here also added the CO2 curve from the same Miller et al. Just to show this drop and to highlight that there has been in recent years there's been a lot of talk about the CO2 variation and orbital forcing being linked, and now such links are also suspected for the Eocene-Oligocene transition. So that this two combined. Here, reconstruction. Here you note that the time scale is the other way around. So this is the greenhouse conditions in the top of the screen with reconstruction of the latitudinal gradient. So the changes in biodiversity across the globe, basically from this hothouse, greenhouse that the Paleocene-Eocene was in the top of the image, where there was a very flat gradient. So there was a lot of tropical forest that extended well into towards the higher latitudes, and this is why, of course, there's famous deposits of like the London clay and the Messel with tropical vegetation, flora, fauna. And at the poles, even temperate to subtropical biota. And then in the ice house, this latitudinal gradient changes, which you see in the bottom of this the series of maps, where there is non-tropical biomes are much more extensive and tropical like biomes are much reduced. And this is a very important sort of red line through the story that I hope to tell you in this time that I've been allocated. So how is plant composition and plant distribution influenced by the EOT? So I've just listed a few things. You can find this in partially summarized in the papers that I've listed below if you want to go back to it. I want to highlight some of the general knowledge on what's happening. So there's a very heterogeneous response to this climate change in the terrestrial environment. And so how is this expressed around the globe? So it's known from North America, for instance, that there was a drop in subtropical broadleafed evergreen forest and the rise of deciduous to conifer forest. And I highlight the conifer forest because I'm coming back to that and highlight some of our own research. In tropical America and Africa, a drop in palm diversity was observed. And we also recently showed that there was also a drop in distribution across the globe. Moreover, Carlos Jaramillo in his work from 2006 shows a very looking at pollen data. And I should say pollen data are very important in reconstructing the biomes, the terrestrial biomes across the globe, because of course they are ubiquitously available. The pollen is produced in large amounts, so that makes them very good fossils to look into plant history. And of course, Bob Spice being here, I also should highlight that not only the pollen, but also the leaves, of course, provide very valuable information on the climatic conditions. So paleobotanical, palynological data are super important to give us insight into this past climate. And of course, also in view of the future scenarios of global warming, the Eocene and the dropout of the Eocene is very important to keep this in mind for future models. Anyway, coming back on what was happening with the plant composition and distribution. So in South America, there's also changes observed in the late Eocene. I think as yet they have not called exactly the late Eocene to Oligocene, but changes from tropical to subtropical flora. In Europe, something similar, change from tropical, subtropical to seasonal temperate. And then Central Asia, and that's what I would like to focus further on in this talk, and also on particular taxa from the tropics. In Central Asia, there's also in well-dated sections for instance, in northeastern Tibet, we find changes from warm, humid to steppe desert vegetation in the Eocene, and then subsequent rise of the conifers towards the boundary. And finally, in Antarctica, strikingly, evergreen forests were reported, which changed to tundra. So this summary is to set a sort of little scene for what can we add to this. And I think also I would like to highlight a paper by Pound and Salzman, published in 2017, where they made a compilation of global pollen records that suggest that heterogeneous pattern of global vegetation change has been controlled by a combination of factors that I listed at the beginning, and that there is not a single response to this rapid decline in atmospheric CO2 at this boundary. So what can new pollen data and more targeted compilations add to this statement that it is a very heterogeneous pattern? And maybe can we draw some more general ideas from these additional data? So first, I would like to focus on the conifers. So what do, for instance, our records from our studies tell us, and how do they complement the existing information? So our data sets that I would like to highlight are from extensive study in the context of a larger program directed by Guillaume Dupont-Nivet on the Tibetan Plateau in the northeast in the Xining Basin. And I would like to highlight a paper by Page et al. That Natasha Barbolini and I participated in, contributing with the pollen data. And so here you see this very well dated section in the Xining Basin, the summary version of it, where you see that up to the EOT there is a presence of evaporites that reflect some humidity in the environment. And then after the EOT, the disappearance of these evaporites, and we interpreted this as being more arid aeolian deposition. And surprisingly, the evaporites provided quite a good pollen record, which allowed us to follow through time the vegetation. So partly the local signal, but also the regional signal. And the regional signal is represented by Pinnaceae, in particular high altitude taxa. And when we first worked on this, quite a few years now back, I think in 2007, 2008, we first found the initial evidence, and now with the other data from that documented in Page et al., we see this transition from at this transition of the Eocene Oligocene transition. This has a very clear increase of Pinnaceae, not only in abundance but also in diversity of types, and it follows very neatly this temperature curve, and so that's a very beautiful indication of the effect that this temperature change has, and we think that is purely the regional vegetation that we're following here, and that the local vegetation is actually comprised by steppe desert taxa, which I'll talk about now. And the steppe desert taxa in this region are nowadays mostly composed by taxa such as the Asteraceae, the Chenopodioideae, Poaceae, and Caryophyllaceae. And so it's that's the characteristic composition of the Central Asian steppe desert. And here's some images from the Urumqi area that I had the great joy to be able to visit some years ago. And but we found that, for instance, in the Xining section, when we look at the total composition, there is not only these conifers at the EOT, but the late Eocene, there is an abundance of Ephedra and Nitriaceae pollen types, which are steppe desert taxa. They're still part of the assemblage, but nowadays they're very rare. And in the Eocene, they were, especially in the late Eocene, they were very abundant. And so this showed us that the Eocene steppe was, well, very different from the present one. And so we suspect that rather than this herb landscape, we had sort of more shrub landscape at the time. And also wondered about the role of the Nitriaceae here. And I would like to highlight here Amber Woutersen's PhD work, which is focusing on the evolutionary history of the Nitriaceae. You see the distribution of this group at present. And these are, they can grow to shrub size. And if you look at the fossil record, part of the composition is of the steppe taxa is formed by the Nitriaceae, which nowadays are known to grow around the salt lakes. They like some humidity. And so in that sense, we think they're different from the, they are susceptible to this change in climate. If we have a change from aridity and drop in temperature, they would respond to that, we suspect. Just want to highlight what is it exactly that we look at in the fossil record when we look at, for instance, these fossils in Tibet. So here you see the Nitriaceae highlighted, the fossil types at the lower part of the image, and the modern ones at the top. And we're also linking these to the phylogeny of the Nitriaceae and see the relationships. And so when you put these local data, these late Eocene local data together, we have this very strong steppe signal that characterizes the late Eocene. So we're coming out of a sort of more mixed humid vegetation, and then this rise of the steppe taxa, but also this steep drop at the Eocene, in the Eocene-Oligocene transition. So we have basically the regional signal of the conifers increasing and the local signal of the steppe taxa declining. And that's what characterizes this boundary in Tibet. And it's combined in this. Here I should highlight that this is work by Natasha Barbolini, who was a postdoc at Amsterdam. And this is a work that was published in Science Advances last year. And where there is evaluation of change through time and contrasting this Eocene steppe with the later Neogene steppe, which is the Neogene steppe is much more like the present steppe. So this big transition, this sort of setting the scene of what the Neogene will look like, is basically happens at the Eocene-Oligocene boundary. And also, sorry, there was, apologies. When looking at data published by mammal specialists, there's also a big change, also known as the Grand Coupure change in composition of the mammals that coincides with this change that we see at least in the Central Asian steppe desert. Just recently, Delphine Tardif tested the Eocene-Oligocene transition made climate models using the combining the CO2 and also modeling the orbital forcing, and what effect would that have on the vegetation. And so I'm highlighting this here because in her reconstruction of the group, they assume that there was a precession-driven monsoon climate in the Eocene, and that the Oligocene model allows for expanded boreal forest. And so if you look at these two images, so this on the left is the Eocene map with more temperate mixed forest in the northern hemisphere, and then you see in the blue here in the Oligocene this extended boreal forest. And we think that the region in northeastern Tibet is part of this. What we are seeing with this rise of the Pinacea, that the model can explain this, that under this changing conditions, changing CO2 values, and combined with the orbital forcing, models explain that there was this extension of this boreal conifer forest. By the way, we also expect that what happened with the steppe, that it was basically a decline, and that there probably was very limited vegetation during this Oligocene glaciation, but it's still the records of the Oligocene are not completed yet in this region, so it's still a bit of a mystery how this how we transition into the Neogene, into this new Neogene steppe. Okay, so now we leave the Tibetan plateau, and I would like to focus on another group, on the palms, which has had our attention in recent years. And the palms are again a group within as a palynological group. They are very abundant, so of course, depending in the palynological assemblages, you're always dependent on pollen producers. So poor pollen producers produce rare grains, but palms are very good pollen producers, so they have a very good fossil record. And they're known to have been widely distributed, originated in the Cretaceous, but also being widely distributed already. This is a map from Vaida and Berkovici, but based on heron grain from 1996. And so you see across Africa and South America, the palm group, the palm province, sorry. And well, inspired by Pan et al., who evaluated the palm pollen richness, diversity, and distribution, and suggested there was a drop in this diversity and distribution at the EOT. We decided to reassess this and see if with new data, extended data sets, how we could fine-tune this further and what else we would learn from the behavior of the palms. And palms are easy to work with, but they're also very important as a model group for tropical forests. So by looking at the palms indirectly, we can also make some assumptions about what's happening to the tropical forest at the time. We focused on two groups, well, on two main groups, the Calamoidae and the Nipoidae. And these are taxa that have a record that goes back to the Eocene and further back even. And the modern representatives are, for instance, Aegisona, Calamus, and Nipa fructicans. And here I refer to Washeng Wang's thesis, who recently graduated at the University of Amsterdam and did a beautiful work on the palms and also on Myanmar. And in his work, he looked at the Eocene of Myanmar. So on one side to reconstruct the vegetation throughout, and here you see a diagram that is the summary diagram where the palms are represented in brown. So he looked at the, well, first of all, at the environments and the composition, abundance of palms in this region, but then also extended further to evaluate these palm species that were found in this record, how do they compare, what was their distribution across the globe in this time. And so what we found, what Weisheng found, was that in the Eocene, these selected palm groups were very widely distributed. So in the image on the left, you see the Eocene with the occurrences plotted for these different palms, and then you can see on the left side their present distribution based on the GBIF data. And just for reference on how do these palms look like, how can we differentiate them, you can see on the right some of the SEM images and the light microscope images. And I should highlight that we had a very nice collaboration with the researchers from Bir Balsani to assess also to compare with the Indian records to get a better understanding of some of these palm groups. So anyway, what we do see is that there is a very big difference between the Eocene and present, and also with further assessments, which are still being evaluated further, it looks like the Eocene-Oligocene boundary played a very important role. Interesting is that in this case, this group of taxa, we see those that sort of retreated to the eastern hemisphere. So we see that the Colpopolis, the Calamoidae, had a global distribution, but then retreat to the east. And this is the same for some of the other groups, Palma pollinitis cuchensis, which is a bit more limited. But for instance, Spinosoma colpitis, the Nipa, was very widely distributed and is nowadays only found in Southeast Asia. And similar for Longapartitis, which is expected to be in Aegisona, which is also retreated to the east. And this is interesting because we move to another group of palms. We move now to the Mauritine palms. This is a group that is part of the Calamoidae and was also evaluated by Weisheng and also by Giovanni, who compared fossil pollen and modern taxa. So this is a group that nowadays is endemic to South America. And here you see an example of a Mauritia palm, Athena, in Ecuador, and they are now very typical for mostly the Amazon drainage basin. In general, they prefer swampy areas, but also have some exceptions in drylands. But they are endemic and mostly occurring in the Amazon drainage basin. If you look at the map that is the result of this vast data compilation that Weisheng Wang did, you see that in the Cretaceous, these palms were reported both for Africa and South America. And this, that in the Paleocene, they come to their maximum distribution. So they extend to India. And then again, in the Eocene, there's already some retreat, possibly because India has moved further north. And finally, in the Oligocene, there's a much more severe reduction. There's still some findings in Africa, but mostly they're already concentrated in South America. So we think the Eocene-Oligocene transition played a role here. And we see in the Miocene, again, Miocene, Pliocene, that it's mostly South America and basically exclusively South America from Pliocene onwards. So we see here this group that was extensive distribution, but now retreats to South America, whereas the other palms that we assessed, they were groups that retreated to Southeast Asia and so on. So that shows that this very important group that are the palms were very severely affected and have a very interesting biogeographic response to this climate change that's happening in the Cenozoic. Just to highlight here some of the images, and this is a very distinct palm group, because I should say palms are not always easy to work with, even though they are very abundant. a lot of them have very simple morphology and they all look alike, but this is not the case for the groups that I've been highlighting here. So I'd like to come to the conclusions now and just list a few things, few learnings from this. So well, our new paleological records from Central Asia and Myanmar contribute to a better understanding of biotic composition from before, during, and after the EOT. And in Central Asia, steppe taxa decline at the EOT, whereas conifers are on the rise. And we also saw how this modeling gave a further explanation how the former distribution would have been. The compilation of the Calamoide and Nipoide pollen shows a notable reduction in geographic coverage of these taxa between the Eocene and present, particularly for the Nipoide and Aegisone. Calamoide are actually quite abundant and common nowadays, whereas Nipoide and Aegisone are more limited in their distribution in the Eastern Hemisphere. And furthermore, a compilation of the Maritime pollen shows a similar pattern of wide distribution during most of the Paleogene, with a reduction at the EOT and subsequent predominance in South America. So they retreat to the other half of the world. And why did the Maritime remain abundant in South America? Well, possibly due to favorable paleogeographic and hydrologic conditions in the Amazon drainage basin. So I think we step by step, palynological work and paleobotanical work can further add to the picture of how the plant biogeography has changed in relation to this important Eocene-Oligocene transition, and how it has really set the scene for our modern world that emerges in the Neogene. So I hope that it gives you a little bit of the picture of the inheritance that the Neogene is from this exit out of the Eocene. And well, I thank you for your attention and wish you a very fruitful meeting. So that's what I had to say.
Thank you very much, Karina. This was a really interesting overview on this Eocene-Oligocene transition, with a lot of crucial aspects that I think we can discuss a bit further now. Are there any questions of the audience? Please raise your hands. Maybe meanwhile I can ask my question.
Yeah, go ahead.
Could you please go a little bit more into this question of the vegetation change on the Tibetan plateau? Because I'm not sure if I got this correct.
Yeah.
You have this profile. In the lower part there is gypsum, in the upper part there is no gypsum, and you say the upper part is drier.
Yes.
Actually,
yeah.
So this was the first thing I thought. I would say where the gypsum is, it should be drier, more arid. And then this lower part is also the part with the nitraria, which is, you also said this, strongly connected to salty soils, right?
Mm-hmm.
And then in the upper part, where there is no gypsum, you have the conifers. For me, this looks like it is getting more humid, and maybe there is more like monsoonal influence or something. I'm curious if I'm totally wrong. So I would actually say the opposite.
I agree that it is very counterintuitive. I think that when if you picture environments in the modern Arabian Peninsula, so there when there's a lot of rain, that's when you get the lakes, and after the rains it basically dries out, and so you see the process happening there. So you see the evaporites forming in the sabkha. And when you see it, when it's raining, it's like suddenly everything turns green and the sabkha is wet. And so this is how you have to envision this environment, and the kind of paleolus that you have there. That's the Aeolian deposit in the drought. And we think so so for instance, Nitraria nowadays, they still grow associated with this sort of sabkha lakes that you have, sabkha type lakes that you have on the Tibetan plateau in certain areas. And we think that these are opposing palynological signals. So you have a local and a regional signal. The conifers don't grow at the same place as the steppe desert. Okay. And so usually the palynological assemblage, you have this local and regional signal. So for us, this represents local and the regional signal. And then on the Ephedra and Nitraria, why do they occur in the late Eocene? This is a drier part. The lower Eocene, the early Eocene, seems to be more humid, and even the middle Eocene is also more humid. So you have a more mixed assemblage with less. So there's not a predominance. There's still the same steppe desert taxa, but it's more a mixed composition. Whereas in the late Eocene they are really dominant. So it's a very unique sequence, I think, in the sense that you have the opportunity of good dating, which was done with paleomagnetics, and this has also been done by the Chinese research groups. And so there's a possibility of very good dating, and there is a palynological signal in sediments where you wouldn't expect such a good recovery. I also would like to add that we don't have a very extensive record of the conifers, only just over the boundary. And that was a very successful sampling expedition from the group with Natasha, that they managed to complete that part, because it's in the list we didn't retrieve so much. I believe Chinese colleagues did retrieve samples, and in fact we have they are in the compilation in the paper by Natasha, where these have been implemented. So from the conifers onwards, then you go into more deciduous forest. Again, regional signal. So I hope that didn't take too much time now.
Yeah, I would like to give the word to Bob Spicer.
So do I need to do something?
No, he just needs.
No, you just have to listen to me for a couple of minutes.
Go ahead.
Karina, very nice talk, very nice summary. I mentioned earlier that the tectonism and mainly fault movements in Yunnan, Red River Fault going into North Vietnam, created a number of basins, and the basin creation is in the late Eocene running into the Oligocene. So potentially we have a number of sections that span this interval. We've only really done work on one in detail. There's Lucha, which has leaves and biomarker records going through, and there's a good magnetostratigraphic framework for it. But, to be perfectly honest, it's a bit boring. We don't see major changes. And as you pointed out, Zaltzman and Pound made the statement that it's very spotty depending on where you are. Topography. That then raises the possibility of lots of refugia and local effects and stuff. And yet we see these broad scale patterns. And I just wondered how, what your thoughts are on going from these local sites where we don't see much happening to these big broad scale patterns, when we have all sorts of genetic reservoirs lurking in these little basins and so on. So how do you put it all together is what I'm saying.
Yeah, I actually think that we actually should put our heads together, because I think there's a number of studies now from the Yunnan area, and I think maybe even also other areas, other places in the region that have an Eocene Oligocene record. So I think we should actually get together and see how we puzzle this together, because clearly there is huge differences, as I know from the valley-shaped topography to, well, of course, the example from Central Asia. So yeah, it's of course, as people say, the terrestrial record is very heterogeneous, and also therefore it's more difficult to reconstruct conditions. But I think by producing more data, we'll get that picture. So I think it's a matter of being patient, work on, and completing the picture.
Okay, good.
And what I think also may help, if I may add that, is that by looking further back in time, we may get examples of what we would see laterally in sort of the later Eocene. So what I'm trying to say is what you see in Yunnan may be something that in Xining we could see in the early Eocene. You see what I mean?
Yeah, it's there's a huge opportunity here, but what I'm really sort of flagging up for everybody, I suppose, is that we get all these nice simple patterns from the deep sea record and all the rest of it, and actually the real world situation on land is far from simple, and maybe we have to think a bit differently.
Yeah, it's far from simple, but at the same time, pictures emerge, like looking at this tropical group of the palms and looking at the arid groups. I think we and with you and colleagues in Yunnan, the picture will become more complete. So.
Well, let's work.
And it's
together.
a fascinating time frame.
Yes.
Thank you.
You can organize a workshop on Eocene Oligocene transition.
Yeah.
Yeah, let's have a workshop.
Vandana, I think you also had a question.
Yes. Arina, very nice talk. I really enjoyed it. Just some query. We get a lot of palm pollen in Paleocene, and it starts from late Paleocene, late Cretaceous to Paleocene. We get a lot of palm varieties. As Nipoides, Camoides, and now we are also finding a lot of Morphological forms. And during late Eocene or Oligocene, majority of the palms, they decline, and then it is like many other forms, dicotyledons, but all these are very tropical families. Your Calophyllum, and then Burseraceae, and then Anacardiaceae, whatever. So it's like a kind of climate change. You feel like from palm dominated to other angiosperm, like dicotyledons dominated evergreen taxa. It's a climate change, or it is what is the driver? What could be the driver? And can we identify the evergreen vegetation into different groups, and can it be called as a like a change in the climate because of change in climatic condition or what? So this is something which is coming up in our research, that many majority of palms which we now see in Southeast Asia, we are getting in Paleocene, but they wiped out during late Eocene or Oligocene from India. Before India,
so their decline precedes really the EOT.
Yes.
Yeah, I remember that when we're working with Bob Morley and yourself on the Maritime, we already faced that issue, and that our hypothesis was that it's because of the migration of India outside sort of the tropical zone, and so that this may cause the decline of the palms, and this is why Mauritia disappears first of all from India. And so maybe that same pattern is continued for other assemblages. So,
but I'm more curious about what was the driver. What was the driver? What was
maybe Bob Morley wants to add to this conversation, right? But please keep it short. We are running out of time.
Yes.
Sorry, can you hear me?
Yes.
Yeah. This really reverts back to Bob Spicer's questions. The petroliferous basins around Vietnam, like the Kulong and the Song Hong, have very complete stratigraphic records and incredibly detailed pollen records, and we've published some of these. And something I would like to see is that localities on shore, some attempt is made to link those with the very detailed stratigraphic successions that we see in these deep marine, they're not marine basins, in these deep freshwater lacustrine dominated basins where we see this fantastic cyclicity. And we see these cycles from lacustrine to fluvial, lacustrine to fluvial. If you saw any one of these outcrops in a small section onshore, I mean on land, it would appear as a discrete, separate assemblage, which should perhaps be confusing. But placed within the framework of these very deep basinal sections that we see in Kulong, for instance, these are studied by the Vietnam Petroleum Institute, but they're very keen to get these data sets published. And so I think it would be very useful to try and tie the records to those sections.
Thank you. I think this was more a kind of comment and not a question.
No, just a comment.
Thank you very much. This gives me the opportunity to continue with our program because we are now a little bit late. And thank you again, Karina.
Pleasure.
For your presentation that raised also a lot of wide discussions. Maybe later we can continue that offline. And I would like to ask Wan Yongjiang, right, to give his presentation on the rise of herbaceous diversity in the southeastern Tibetan plateau. Maybe this is linked to the topic we had right now. Please share your screen. Let's see.
Oh, sorry. Yes. Oh, and yeah, I can't open the link.
You cannot open your presentation?
No.
Oh.
I click the share, but it does not work.
Oh.
Can you send your presentation over to BSIP and we can present on your behalf?
So you can send your presentation to the organizers by email and they can present it for you?
I contact the organizer and but I already opened the link and I can hear from you and I can watch your PPT. Can you see my PPT, the slides?
No.
Mm-hmm.
No, we can see you as a nice picture, moving picture, very smooth, but we cannot see your presentation. So have you, what happens when you press the Present Now button at the bottom of your screen?
Well, I pressed the button of share, but it does not work. It's gray in color.
Maybe if you try and come out of Google Meet and come back in again.
Oh, does it work?
Is it appearing now?
Okay, so shall I start now?
Ah, yes.
Yes, good.
Something is happening.
Good.
Cool.
But we're seeing your screen, not the presentation.
Yes, thank you, Angela. Can you watch the slide?
No.
Oh.
We see your browser.
Yeah, we see your browser. So Google.
I think you have to unshare and share your PowerPoint or your full screen or something.
will do this. Minimize the browser.
Fang, just minimize your screen. Fang.
Yeah. So now can you see it?
Yeah.
No, we only see the browser.
Minimize the browser.
Young, do you have multiple screens, or have you just got the one screen?
I'm using a Microsoft Edge, but sorry.
We just got a white screen just then, which showed something was happening. Now we see you in the lab.
Dr. Huang, maybe you can open your VPN so that it can work. If you have VPN, please.
Can you see it now?
No.
Sorry. It does not work.
Something's happening now.
Yes.
Your browser again.
Shall we move to next presentation, and I will talk later. And
yes,
the problem is so.
Thank you. I think we. Sorry for that. So please unshare, and we. Yeah, maybe you send your file to the BSIP, and they will present it in your behalf while you are talking. Meanwhile, maybe we can try the next presentation about giant gastropods of the Miocene of Dwarka Basin by Kanishka Bose.
Yes, let me share the screen.
See if this is working. Yeah, something is happening.
Okay, is it visible?
Wow,
my
yes,
presentation?
perfect.
And I'm audible? So...
Yes.
Okay, let me start. Hello everyone, I am Kanishka Bose from a research scholar from Indian Statistical Institute, Kolkata, and I will be speaking on two giant gastropods which we have found from the Miocene of Dwarka Basin, Gujarat, and we were discussing about their paleobiography implications. So now let's talk about the Dwarka Basin. It's present at the westernmost fringe of the Kathiawar Peninsula of the Gujarat. And the Miocene succession overlies on the, it's a pericratonic basin, and the Miocene succession overlies the Deccan Trap, and the Miocene succession can be divided into two formations, namely Kutch formation and Dwarka formations. While the Kutch formation, this stratigraphy is given by Jain 2014, and the Kutch formation can be further subdivided into 7 members, while the Dwarka formation is subdivided into 3 members, of which our focus is the entire Kutch formation, and this Rangasar member of Dwarka formation. So now we talk about these two giant gastropods. One of them belong to the genus Dilatilabrum of the family Dilatilabridae under the superfamily Stromboidea, while another genus is another gastropod belongs to the genus Lithogonus, belonging to the family Coneidae. These gastropods have been observed from Western India in past by several researchers. However, their similar morphology and lack of well-preserved specimens created confusion, thus resulting in lack of detailed study on them. These giant Kona species, Lithogonus gajensis, was reported by Jain in 2014 from Dwarka Basin. However, he did not give any proper systematic description, and also his collection was very limited, both in spatial and temporal terms. And however, he indicated the presence of these giant stromboid gastropods, which were embedded in the rocks at internal molds, which gave false impression of the lithogonous gazensis. Thus, we need to do a detailed taxonomic studies on these two taxa to avoid potential misidentification in the field. Now, coming to the...
Excuse me, may I?
Yes.
May I quickly interrupt you? Is everybody seeing the presentation now?
Yes.
Because
Yes.
in the chat.
No.
Yes.
Yes, we can see.
Can I continue?
I've just got a black screen.
Yeah, but I think everybody else is fine.
Yeah.
But maybe
Well, it was doing fine until just now.
maybe you have to go out and come back again. No, Kanishka, Bose, it's all okay. Everybody except Bob Spicer can see your presentation. So.
Okay. I
Sorry.
think it's the problem in his internet, I think.
Yes, please continue. Sorry for interrupting.
Okay, so again I am showing the map of Dwarka Basin, and these blue diamonds are the fossil localities from where we have collected these giant gastropods. And this is the lithostratigraphy succession, where as we can see that the giant conus, that is lithogonous gazensis, have been collected. We have collected from Dandana, Kuranga, Ranjitpur, Laurali, Davla Mandir of Kutch, and Rangasar member of Dwarka Formation, while the giant stromboid, that is Dilatilam SP, have been collected from the Ranjitpur member and Davla Mandir member. So these are the gastropods collected, Dilatilam SP. First, let's discuss about this genus. Dilatilam is an extinct genus belonging to a family Dilatilabridae, which were mainly confined to the Tethys region and earlier had a range from middle Eocene to early Miocene, that is Aquitanian. This genus is a direct descendant of the genus Ortholox of family Thercetidae, and which is believed to be the ancestor of the family Strombidae. And morphologically, this genus is marked by characteristic smooth shell and a prominent sharp keel with a winged aperture. So they were mainly reported from, as I said earlier, they were mainly reported from central Tethys, that is Meteriac and central Iran. And a single occurrence from late Oligocene Chathian is reported by Harzhauser et al. 2009 from the Kutch Basin. This one is our species we have collected from the early Miocene and middle Miocene of Dwarka Basin. So here, as you can see, this is the geographic distribution of the there are 4 species reported till now, including ours, and these are the geographic distribution in the present-day world map. And these are the paleocoastline maps of Eocene, Oligocene, and Miocene showing the distribution. As we can see, that the blue one is Dilatilam fortici, which this genus originated during Middle Miocene, and the distribution was seen mainly in the central Tethys, western and central Tethys, and a single occurrence in the western India. However, during Oligocene, the genus become much more widespread and abundant, with 2 species reported, that is Dilatilam roigli and Dilatilam sublatissimus, which were which had an extent up to western the western India in the easternmost part of the Tethys. However, during the late Oligocene, we can see a migration of the genus towards further east. This occurred due to a significant cooling event which persisted in the Mediterranean, and they thus migrated towards the this Indo-Western Eastern African province, and thus the early Miocene occurrences were mainly reported from Somalia and Tanzania. Our species Dilatilam sp have demarked its It's up to the we have found from Dorka Basin. It's up to early Miocene to middle Miocene, which thus extends the last appearance data of the genus up to middle Miocene. Also, it demarcates the easternmost extent of the distribution of the genus up to the Western India. So now talking about the association, this genus is mainly associated with gastropods like turbinates, cassis, and less frequently with potamerids and naticid gastropods. A similar association were found from Ranjitpur and Dabla Mandir members. And paleoecological analysis of the genus by Herzhauser revealed that this genus is mainly found in warm, shallow marine environment with sandy bottom and scattered patchy reefs, which is typical strombite paleohabitat, and similar substrate can be seen in case of Ranjitpur and Dabla Mandir member, which tells us about the environment when these gastropods existed. And construction and morphological studies of stromboid gastropods revealed that the epifaunal locomotion of stromboids were in leaping fashion. Means they would jump when they tried to escape from predators. However, when they had to land on inclined substrate, they had a high risk of overturning and thus were vulnerable to predators when they tried to right themselves. So having winged aperture, broad wing aperture or set of spines around the aperture increase their footprint of landing, like in the case of snowshoes, which are used by while doing ski in snow. So these representatives of Dilaterion did acquire a broad wing aperture and a thick shell to prevent as a preventive measure to avoid predation. However, their large size and smooth ornamentation might be a possible reasons for the extinction of this group. Now coming to the giant conus species, Lithoconus gazensis, its maximum height we have obtained is 23 centimeter, and these are the some photographs of the specimens. This is comparable to our recent species Lithoconus leopardus, which is one of the largest conus in modern seas, and it's also has size range up to 5 centimeter to 22 centimeter. This Conus, Lithoconus gazensis is only found from the Miocene of Dwarka Basin, and have not been no giant Miocene conus has been reported in the world so far, thus making this species an endemic species to Western India. I would like to conclude this talk with and acknowledge Indian Statistical Institute for logistical and financial support. I would also like to acknowledge ACRB DST for the financial support. Thank you.
Thank you very much for this nice presentation of beautiful gastropods. Are there any questions? Can I see? Hannah Zuba.
Yeah, so hi Kanishka, nice presentation. Yes. I just wanted to ask you, how well preserved is the calcitic part in the shells? Like, is it
but most of the specimens are internal mold. However, we have found some parts of the cells preserved in few specimens.
Because why I'm asking is, like, in case we need to do some stable isotope or clumped isotope studies, like I think I don't know. I just wanted to know if we will get any signature of diagenetic alteration that would be there.
For few specimens, there is shell preserved, so you can definitely do stable isotope studies. However, most of them are internal molds.
So right now, none of these species that you have found have been studied for stable isotope.
No, none of them have been studied. The basin is totally underrepresented in the literature.
Yeah, because this was a new species, I think. I mean.
Yes, I haven't reported in the literature. Yeah. This is prior to that.
Thank you.
Yes.
Thank you also from my side. If you don't mind, I would like to continue and give Juan another try for sharing his screen. We can go back to herbaceous vegetation of the Tibetan Plateau. Good luck now. Is it working? Yeah,
Hi, Angela.
No, there's
Now, can you hear me? Now, can you hear me?
Yeah, can hear you now.
There was a feedback. You were sharing the meeting, so there was an endless feedback. It was a very loud noise. That was all we can see. Did you send your presentation?
We have received Huang's PPT, but somehow we are not unable to download it. We are still trying to get it downloading up and running.
Okay, then last try.
Okay.
We are just trying to open his PPT.
Otherwise, we will continue. I suggest to continue, actually.
Okay.
We can continue to try.
Oh, here. I found it. Can you see the slide? Angela?
We have to wait. Yes.
Yes, it's coming up now.
Oh, so shall I start now?
Okay. Yes, please.
Well, I'm so sorry for the delay. Okay, good afternoon to the colleagues from India and Nepal, but good morning to our friends from European countries and the US. My talk is the rise of herbaceous diversity in southeastern Tibetan Plateau. First insight from fossils. And firstly, I will give a brief introduction of the background. The Hendan Mountains region, shortened as HMR, is located in the southeastern edge of the Tibetan Plateau and also constitute the southeastern part of the plateau. And this region is diverse in environment because the altitude increased from below 1,000 meters in the south to above 4,000 meters in the north. So it has high diversity of ecological niches. So it is home to extremely high diversity of plants. There are more than 20 families of seed plants in the region, presenting more than 70% of the total families in China. And also it is one of the 25 world diversity hotspots. And due to the high altitude and temperate climate, the Hendan Mountains is particularly diverse in herbs, and it represents the centers of many herbaceous groups, and many of these herbs contribute to alpine and subalpine meadows and the grassland. So how the diversity of herbs is established? Our current understanding is based merely on the study of modern plants using the molecular phylogenetic analysis. These studies indicate that the many herbaceous groups experienced rapid diversification, mainly during the Miocene and also afterward. However, the fossil evidence demonstrating the rise of hops is still lacking. So in this study, I will present a fossil locality that produces many fossil fruits and seeds of hops. The fossil locality is in the Kutch Basin, and the age of the sediment, the fossil-bearing sediment, is late Pliocene. And we have collected a large number of fruit, seed, needle, and charcoals. And in this study, we mainly focus on the fruits and seeds of herbaceous plants. And we totally identified at least 18 species belonging to 11 genera from the fossil collection. And the number of species is still growing because we are still continuing to identify the remaining fossils. And these genera include Raniculus and Corydalis and Rumex, and most of them represent the first record in the region or even in the whole country. And I will quickly present the SEM images of the fossil fruits and seeds. And these are two different species belonging to Corydalis, but the species assignment is difficult because firstly, the genus is species rich, and also because using the seed morphology alone, it includes only a few taxonomically important characters, but the genus placement is quite convincing. And these are two species of Polygonum, and the seeds of Polygonum is quite characteristic because it has two surface types. One is one type of surface is reticulate, and the other is smooth. And here is the two species of Stellaria. They are very unique in the surface sculpture. And this is Astragalus, but the species assignment is left unresolved. And also other genera of hops. And the Calyx seeds is species diverse, and I recognized at least 5 species, but it's difficult to assign them to a specific species. And also the Sconoplectus. This is an aquatic to subaquatic plant living in the wetland or close to the wetland. So there are a considerable high diversity of hops in the fossil assemblages. And back to the background, as I mentioned, in the Hendan Mountains, many herbaceous groups have experienced rapid differentiations during the Miocene and also the Pliocene. So we think the local assemblages of these hops in the fossil site might be the result of the diversification of many hops. And we looked at the habitats of the hops, and we found most of them today live in urban environment. For example, the forest edges, the mountain slope, the meadows, the grassland, and the wetland. So we consider we consider an open meadow vegetation probably occur in the basin during the sedimentation. And we also look at the contribution of each herbaceous element, and we found that the polygonum is quite dominant, followed by astragalus in terms of fossil specimens. So we think this fossil meadow or wetland is probably dominated by these two plants. And today, the meadows dominated by polygonum is quite common in Hendan Mountains. And here I show the field picture of a meadow in the mountain slope. But this meadow is probably disturbed by human beings. And it's not nature, but we believe such kind of meadows naturally exist in the region today. And in the Hendan Mountains, there are many. There have been many fossil floras, but most of them are composed of leaf fossils that represent woody species. So they may represent forest vegetation. For example, the broadleaf forest, mixed conifer forest, and the sclerophyllous forest. But all fossil finding indicates the first urban vegetation, the meadow vegetation. So this will enrich the vegetation diversity in the late Pliocene of the region, and also sheds light on the formation of the modern vegetation diversity in the region. And finally, we look at the causal relationship for the formation of herbaceous diversity and also the rise of meadow vegetation. Firstly, we think the temperature decline in tune with the global cooling is quite responsible, because among the hosts in the fossil assemblages, most of them are temperate in climate preference. And also the Hendan Mountain experienced a severe uplift caused by the uplift of the Tibetan Plateau. So this surface uplift may intensify the temperature decline. And these two factors may lead to the rise of herbaceous diversity, particularly those temperate types. And here is a conclusion. First, our study provides the first fossil evidence showing demonstrating the rise of herbaceous diversity and the formation of meadow vegetation in Hendan Mountains. The local assemblage of these hosts in the He Basin may be resulted from the diversification of many herbaceous groups mainly during the Miocene and also the following periods. And finally, temperature decline associated with the global cooling and regional surface uplift is probably the major driver for the rise of hops and meadow in the Hendan Mountains. Thank you for your time.
Thank you, Huang, for this nice presentation. So after the difficult start, this was really beautiful to see this fruits and seeds flora. Are there any questions from the audience? Actually, I have several, but I would give you the opportunity to ask something. No, I do not see. Torsten. Yes, Torsten has a raised hand.
Yeah, very nice contribution, beautiful materials, thank you. I would have a question related to the type of flora you analyze. I mean, in the fruits and seeds, herbaceous vegetation is overrepresented anyway. And to my knowledge, there are not so many fruit and seed floras in this area which are really well studied. So maybe we should be more careful with the conclusion that this diversity evolved in the late Pliocene. Maybe you would find early Pliocene or late Miocene floras when you look more closer with respect to carpology, you know, and then might detect a better diversity of herbaceous species as well. What do you think?
Yes, thank you for the suggestion. Yes, in the Hentai Mountains currently, there are many fossil floras, but most of them are leaf fossil floras. So they reflect mainly the diversity of woody plants.
Yes, but there is also this taphonomic effect that in leaf floras mainly more robust arboreal taxa and shrubs are represented, and only herbs are notoriously underrepresented.
Yes, and to date there are a few fossil floras composed of fruits and seeds. And fruits and seeds, they will represent both woody and herbs. So maybe in the future we will find more such fossil assemblages to more closely reflect the past plant diversity.
Yes, I agree. Yeah, but nice contributions. Thank you.
Thank you.
Yes, I agree. And maybe I can add that this study shows how important it is to look at all kinds of plant fossils, combining leaves and seeds and pollen, of course, which can add to this discussion. Yes, I thank you again, Huang, and I would like to proceed with, if I am correct, we are now going back to the Kutch region in India, right?
Okay.
And yes, we will.
Let me share.
We will hear a presentation about amphibians and snakes from the neogene localities of Kutch, presented by Nongmai Amada Singh. Is it correct?
Yes.
Please try to share your screen.
Is it visible?
It's coming.
Okay.
Yeah, we see the PowerPoint. I think you have to.
It's now in full screen or what?
No, it is not. So maybe you have to use this publishing view or what? No, you do not have that. Okay, may we get some instructions from the organizers?
No, Maxim, can you do a full screen, please?
Yeah, I have done full screen, but
It's
in my
not,
screen it's full screen.
It's not working again.
Can you click on that book button, which shows
It does not
overview?
have.
Yes. Click on that.
This one is slight shorter. This is not. Just
No,
I keep this.
He does not have this. It's an old PowerPoint.
F5. F5. Can you try F5 key?
Yeah. But in my screen is full screen. But
Yeah, probably you shared the wrong screen.
so can I start like this?
Yes.
Yeah, it's coming now.
Okay. So my name is Amadas from Central University of Punjab, Bathinda. Today I'm going to talk about fossil amphibians. and is next from Neogen locality of Kutch, Western India. So come to the Kutch. Kutch is the largest district in state of Gujarat, which is located westernmost part of the Indian subcontinent. So geologically, Kutch is a pre-Cretacic rift basin which preserve almost a complete sequence of sedimentary deposit ranging as from Triassic to recent. So Miocene locality, we know that it's famous for rich assemblage of mammals and other vertebrate fauna. But some about the amphibian and reptile, we know very little about this fossil from the Miocene of Kutch. So most of the amphibian and reptile reported from Kutch are from Eocene deposit, and Miocene record are very limited and find in very few places. So recently we have collected some amphibians and snake fossil from two different locality of Miocene Kutch, the middle Miocene deposit of Palasava and the late Miocene section of Tapar and Pasoda. So most of the recovery fossils are fragmentary, which make taxonomical identification is very difficult. However, unfortunately, few elements, amphibian and snake, are identifiable at the generic level. So this map is the geological map of Kutch, modified after the GSI 2012, and this star mark is showing the fossil locality. The one is in Tapar and Pasoda, and this one is Middle Miocene Palasava. So left side image is showing the litho section of Tapar locality, and right one is the litho section of Palasava locality. And the green line is shows the boundary of Miocene deposit of Kutch. So these are the fossil ilia bones of frog rana. And so this first figure is the fossil vertebra of frog rana, and middle one is the vertebra of Renidia, and last one is the vertebra of Anura frog. So, and these are the humerus bone of frog Renidia, and below one is the humerus bone of frog Anura. So come to the snakes. These are the vertebrate bone of snake python, which we have collected from the Palasava and the Pasoda. So this one is the vertebrate fossil of snake Acrocotus demi. The upper first figure is the Acrocotus demi, and the lower one, below one, is the Acroconus sp. So, and this one is the snake vertebrate of Helptunilia, and lower is very fragmented. So this one is questionable. So, but we keep this in correlate indeed. And this map shows the Neogen occurrence of this fossil rana, python, and Acrocotus. So in discussion, first paleo environment. The late Miocene deposit of Tapar and Pasoda locality shows the freshwater environment based on the presence of rana, which is supported by previous report of various geologic fossil remains. However, the presence of such python and Evolutionary indeed represent the open terrestrial habit. This and the occurrence of reptilian and numerous mammal fauna comprising turtle, crocodile, squid, squid, and rhino, giraffe, proboscidean, etc. shows that Tapar might be wet and humid climate condition during that Miocene, and which is also supported by the stable isotope data by Panaik et al. 2019. And come to the Palasava middle Miocene. Palasava locality is shows that the prevalence of woodland forests close to freshwater and marine environment based on the occurrence of freshwater fish, and some mammals by Kapoor et al. 2019. And the addition of this herpetofauna assemblage, the Palasava locality also indicate the existence of humid climate condition. So conclusion, the species we have described here is identified as Rana sp, Renedia, Anura, python, and Acrocotus demi, Acrocotus, and Evolutionary. And the specimen Rana and python is the first from the Miocene occurrence of taxa from the Indian subcontinent, and this is also the oldest record here. And the presence of Palasava study here indicate the warm, humid and tropical to subtropical environment condition. So, thank you.
Thank you very much for this interesting talk. Are there any questions or comments? Or may I ask you, where are you? May I ask you to see you?
Okay, I'm presenting from desktop, so my...
Okay, no problem, but you are still there.
Yes, yes.
Isn't Rana a frog that is living everywhere?
Yeah,
Is it with wide distribution globally?
Yeah.
So I think it's difficult to infer about the climate from that.
Yeah, but we have collaborated with that stable isotope data and mammals. So they are reptilian are ectothermic. So from that we had just.
Yes. No, I just think you may be a little bit more careful to say, okay, your interpretation about the climate is based on all the fossils. And not really on the amphibians and reptiles, right?
Not only on the amphibian. I didn't follow.
Yes. Okay.
Yeah,
Thank you.
May I? Can I ask a question, Angel?
Yes.
Please.
Nongmetham, very nice presentation. Just a query from my end. Palasava, we already know that we have like marine elements as well as non-marine elements in there. But in Tappad and Pasula, I'd like to know, do we have any marine elements, anything you've come across?
Not yet.
Not yet as of now.
Yeah.
Okay. Oh, okay. Thank you.
Oh, thank you.
Yes. Thank you very much. Let's move on to the next presentation by Ramandat Sagar. Is there? Preliminary data on coprolites from the Neogene. Again from the Kutch region, and I think there is a. I don't know. See. Feedback again.
Yeah, it's visible, Ramanand.
Yes, sir.
It's visible, but with feedback.
Nice. First slide.
It's a system problem.
We cannot hear you.
Hello.
रामानंद आपकी आवाज कम आ रही है
हेलो
सिस्टम की वॉल्यूम बढ़ा
हेलो सर for mic I have joined through mobile hello
okay
hello I'm audible
very low voice and there is still the feedback
hello hello
yes say something
hello am I audible
well it's very low voice I do not know what to recommend Is there any technical support available somewhere?
Yes. We are trying. He is
Thank
from
you.
our office, so we are trying.
Okay, we have a nice view now on the presentation.
Yes.
Hello.
Oh, yeah, I think if you speak a little bit louder, it will be okay.
Hello.
Yes?
Hello.
Yes, we can hear you saying hello. Maybe you speak a little bit up and can start.
Can I start?
Now you have the feedback again.
Hello.
What is this?
Hello. Am I audible? Now I'm starting. My topic on the preliminary data on coprolites from the Neosine, Miocene, accurate age is Equatorian and Burjgalian, Kharidi and Chasra formation from the Kutch Basin Western region. Actually, it is I can say it is a new findings. So that's why I am here presenting the only preliminary data. Before going to my data, I want to present some timeline of coprolite research in India, because coprolite research is not new. It started from 1859, but still new in India. Not much of the research have done in this field. Only, as you can see, only these numbers. There are numbers I have reported. I have mentioned here that was reported in India. First time coprolite was reported from 1859 by Oldham. Actually, it was collected by in 1857, but he documented in 1859. 1859, and then again not any study have done on coprolites from 1881 to 1935. Again the confirmation of the coprolite in the Triassic deposit of Mallory formation. Actually first reported by Oldham, that was also from the Triassic deposit. Again then it was the Matley who have discovered and who gave detailed study about the coprolite from the Mallory formation of Triassic age, and also from the late Cretaceous diameter formations. And according he gave detailed study of the classifications of the coprolites. Then again the gap of 36 years. In 1975, it's come with new coprolite formed by Sani and et al. It's time of Eocene time. Eocene time. Then again in 1929. In 1979, the coprolite found it was give new inclusions, biotic component ostropod that found within the coprolite. In 1939, 1983, and 1989, we found in Indian researcher found Jurassic coprolite, but not of detailed study. A detailed study has given. From there, from 2001, the study is coming day by day. As we can say, the study coming in frequent years, as you can look here. And lastly, in 2020, two more new coprolites, I can say. From one is microcoprolites. Before that, all are coprolites. In 2020, it's a microcoprolite, and another one from the Raksit and et al. That was coprolite of sharks, scroll coprolites. That's also new evidence from the India. Moving to this, this slide, I have put all the places from where Indian researchers have found the coprolites. till later. Still, someone maybe missed, but these are the first on Malari formation from the reported, and up to present time, whatever research have done in India on coprolites, most of was emphasized on late Cretaceous Maastrichtian time type of coprolites. That was herbivorous dinosaur. Moving to next one, I have plotted here Kutch region map. Uh, it is for two reasons. First, to locate my locality. These are my locality from where I have our sample was collected. First one is Matanomard, Kirsara, Palasawa, and here I have quoted sentence. I think it's going true because as we have seen many lectures from the Kutch Miocene region, Kutch Miocene region is getting highly importance because same time sequences of Sialic region and in Kutch region have the larger number of abundance number of the vertebrate fossils, but in neogene of the Kutch region recovering both terrestrial as well as marine vertebrate fossils. That's why getting more and more significance. But I am here to present about the coprolites. I can say that, however, no or little emphasis has been given on coprolite research. Moving to next one, I have here put some photographs from different regions. This is from Palasawa. This is from the Kirsara formation, and this is from the Matanovar formation. All of the coprolites recorded here were recovered by surface prospecting method from the outcrop. Outcrop we collected from surface prospecting method. Again, here I have put the digital photography images here to show the collected samples of the coprolites of both sides of these samples. I put it here. All our lateral views. In this slide, I have to focus. I'll tell you to focus on this sample, A3 sample, because in this sample there is ornamentation over these. These patterns are called rib patterns, generally produced by the Chelonian coprolites. Again, this slide, total of the nine coprolite specimen, whatever we recovered, and picture of both sides. One more sample I'll tell you about. This species, G1 species, if you look in this part, here shark teeth is included with sediment. It is not included in coprolite. It is included with sediment, but that's why you can say at the time of the deposition, shark livings and shark teeth were also associated with that. Moving to that, I have presented here the size comparison because up to present study for coprolites, size comparison is important because in their classification, not a certain classification have been given to classify this coprolite belong to this coprolite to belong to this. This is only the size comparison through people interpret that this belong to this may be this, and here saying. In this plot, I have plotted Miocene coprolites, total Miocene coprolite, whatever recovered in India. These are from the micro coprolites from the Miocene Basin, Kutch, and I have also plotted some Miocene coprolites from the Mukurmatia Baripada beds from the Odisha region. And my sample is falling in this region. Group 2 of the Baripada, Mukurmatia collected samples. So here also through this interpretation, I can also say that my sample is lying in association with crocodilian sample that reported from the Miocene of Baripada Basin. Again, size comparison sample. Here I have put all the Miocene sample as well as the all previous samples recorded in India. For in this slide, I tried to that beside the myosin sample assemblage, my is assembled to any other sample that recorded earlier. Um, in this interpretation, my samples, these elliptical myosin and irregular myosin that I categorize, falls below to type BA late Cretaceous young that's belong to the producer young Chelonian small size dinosaur, and also lying assemblage with the type B Cretaceous Maastrichtian that's Chelonian. Here this is the sample ribbed one that's mostly incorporates to this Chelonian small size dinosaur. Moving to this, after the direct physical appearance of the sample, I have used some instrument image, SEM image. This is the SEM image, and I have tried to put the image from the matrix and inclusions. This first one is the image of matrix of the coprolites that so clearly of the containing or the composed of the calcium phosphate. That is the characteristic composition of coprolites. I have taken 4 points here. In every points, the calcium phosphate is dominating. That's prove. That's prove, and that's give the idea that if the sample collected is calcium phosphate or coprolite. Again, in this slide, I have put the same SEM image again, and here I have tried to put inclusion of the coprolites, inclusion that present in the coprolite that produced by some taxa what they have eaten. Here is the inclusion of the bone matter. Uh, in this picture, I have taken 5 different points, and every 5 points showing the calcium phosphate. That confirm that this is the bone. This thing is bone matter, and the matrix surrounding this showing the calcium phosphate, showing the coprolite composition. Again, I have put a same image. This is probably egg cell that generally we found in lots of in coprolite. Some foreigners have reported in egg cells in coprolite. I might be wrong, but again here I have taken 5 points to show the really these points or these composition these sample showing the calcium phosphate or not. Every point is showing prominent calcium phosphate. That's so this is coprolite. Again, for the more evidence for being a sample being coprolite, I put it 8 more images. First is this. This is general characteristics of the coprolite that for our coprolite, it should be a gas bubbles like a structure, macrospletic like a structure that shows clearly here. The macrospletic like structure are present. That's formed due to the bacteria that formed due to bacteria's work. And in image, again I have tried to show something more that it prove that its sample is coprolite. Here is a pyrite crystal and also surrounding the macrospletic structure that prove that these samples were in reducing environment conditions. Again, in this F image, again the bone matter that was preserved in the coprolite specimen. In G image, also the coprolite bone matter that preserved more better than others, and also showing the macrospletic structures. In next one, again I have tried to show more inclusion that were in the coprolite samples. This is the bone matter that clearly preserved. Again, this is the bone matter clearly preserved. This one is also. And in K image, it may be the secondary mineral matter. It's showing calcite also, but it's not a bone matter. Moving to next one. I have tried here to again plot the all the sample with their dimensions in PCA plot that gives the combination, or that gives the differentiation, how to differentiate or how to assemblage the samples with older ones. I have used 2-dimension width and length width ratio. In this region, in blue line, our sample is falling. And in this yellow line, whatever the crocodilians or chelonian samples of older samples recovered falling in this sample. That's through this PCA, and also I've showed earlier size comparison slides. I can say that my samples are falling in association with crocodilians and chelonians. Moving to next one. This is the conclusion one. For conclusion, I have to go for second point first. Actually, my samples were the new findings, so I have to prove that these were the samples belonging to coprolites or not. From my data collection, it's proved that it's coprolite, showing the same image from the same ADS data. It's have the bone matter, it's have the calcium phosphatic compositions, and because of including it includes the bone matter, the coprolites belongs to the carnivore diet. Carnivore diet means the animal that produce that coprolite are the carnivores. Again, moving to third point, morphometric and size comparisons, whatever I have done in PC analysis and size comparison, normal size comparisons, that's feed with Mucromatia samples, and also with the small size late Cretaceous, a small size dinosaurs and chelonians. Through this, I can say that my samples belongs to crocodilians. And last one is first one, is important one, because coprolite studies, as I told you, coprolite studies is not new but still new, because first coprolite found from Triassic succession, but still not in every time sequence we found. We are feeling the gap of Miocene age, because we found the Triassic, Jurassic, Cretaceous, but not in Paleocene. We have the Eocene sample. Now I am providing the Miocene samples to fill the gaps, fill the time sequence of the coprolite research. Thank you.
Thank you very much. There is already a note by Vijay Prakash Mishra,
congratulations to for this coprolite very much studied in India.
Excuse me. Somebody has to mute the microphone.
It seems so. So just wait.
Ramanan, mute your microphone.
Hello. I hope.
Yes, now it works.
Now it's good, perhaps. So congratulations to Dr. Ramanan for these studies on coprolites, not too much studied in Indian context. As far as the gap, he showed that right from 1940 to 75. The 75 paper was myself with Dr. Sahani in 1975 on Eocene coprolites, crocodilian from Kutch. So, but in between, there are records of coprolites from Shivalik beds in India, from both from middle and upper. There is a paper by Dr. K.N. Prasad, his Paleontologia Indica, and on Haritalianga. So Dr. Ramanan may see that paper. I also recorded with our colleagues, with Mr. Verma and others, way back in 2002, Shivalik coprolites of quite large sized. Those were quite large sized. Those are displayed in the Shivalik Fossil Park at Saketi. So there are a lot of records from Shivalik sediments also in India. It is to the data of the Dr. Ramanan. Congratulations to him. Thank you.
Thank you for this comment. I think we have some sound problems with Dr. Ramanat, but I would also, maybe somebody can answer this question for me. There are more animals available except crocodiles, right? And I'm wondering if, what about hyena? Is it a potential source of that coprolites?
Can I answer that, Angela? Dr. Angela? Yeah. Okay. The first thing is we wanted to here only show the preliminary data on coprolites. If you see the last 150 years of research on coprolites in India, most emphasis has been only on the type A coprolites, that too linked to titanosaur dinosaurs, herbivores, from the Maastrichtian of peninsular India. Now, why that was done? Because people are very fascinated with dinosaurs. So people started looking into the diet of the dinosaurs. Rest, all the other coprolites which were linked to rhynchosaurid reptiles or any other groups, were never, nothing was seen in situ for those coprolites. Even for the Shivalik coprolites or equivalent Neogene coprolites or Eocene, Middle Eocene coprolites from Kutch region, which Dr. Mishra just mentioned, never ever has an in situ work has been done on these type of coprolites. That's one thing. So it's in a preliminary stage. There are not one but many groups of animals which have been reported from the Kutch Basin. You have already seen in two of the presentations today by Singh and Anusuya Bhandari earlier in the previous session. And it's very difficult as of now to, because there are n number of problems here. First thing is the classification scheme which we are following was the initial classification scheme was given by Matley in 1939, and that's what everybody followed. And after that, very new coprolites which are coming from Europe, from the I believe from the Neogene of Europe. Di Pedro et al. Is a paper in 2017, where they have provided some classification schemes for those. We tried to follow those classification schemes. So morphologically, again, sometimes it's difficult to put that particular coprolite in that particular shape because there are in numerous types of shapes. Now, the third thing is. Why crocodiles? Because the only other coprolite material which we have from India where we can compare with the size of the coprolites or morphology of the coprolites have been linked to crocodiles only. And another reason for that is in case of Palasava, they have been all these three areas, Palasava, then your Matanomat South locality, which is Kotada, and then your Kirsara. All three localities we have found crocodiles associated with them. So that's how, as of now, the hunch is that they are crocodilians, but time will tell. This is the first time ever we are seeing bacterial molds in coprolites, in any Indian coprolite. Before that, nobody has reported such things. There are soft tissues which have been reported, like mammalian hairs and all. In Europe, coprolites, probably the preservation is much better. None of that has been reported in India so far. So it's difficult as of now. I hope you understand, but work is going on. So let's hope. Yeah.
No, I think this is an interesting path to follow, and I'm looking forward to further reports or publications from your side.
Thank you.
And now maybe we go back to fossil plants and plant biogeography, presented by Haposhi Hazara on diversity of Engelhardia from the latest Neogene of India. I hope we can get this. Already strange sounds.
Good evening. Am I audible to you, ma'am?
Yes, you are. And we see your slides.
Good evening, everyone. Myself, Tapushi Hazra, Research Scholar, Department of Botany, SKU Purulia. I am here to present diversity of Enceladina from latest Neogen sediment of Eastern India and its biogeographic implications. So, here I begin. Enceladina, an endemic genus of walnut family Juglandaceae, and is considered to be the most primitive genus. The genus is widely distributed in tropical as well as subtropical forests of Southeast Asia. The genus is represented by 5 species, of which only one species is found in India, that is Engelhardia spicata, which is confined only to eastern Himalaya. However, the subfamily Engelhardiidae have 3 genera, that is Engelhardia, Aurumonia, and Alphora, of which the Engelhardia is confined only to Southeast Asia and Aurumonia and Alphora is confined only to Central America. The Engelhardia bears a typical trilobed wing fruit. And next, I come to the fossil history of Engelhardia. Here you can see I have presented a table showing the various earlier fossil record of Engelhardia, of which the earliest fossil record has been recorded from Mississippi, USA, that is of Eocene age, followed by Oligocene from Armenian France and from Guangxi, China, Niming, China. And the Neogen record have been reported from Yunnan of China. However, unfortunately, there is no record of Engelhardia from India. In this pretext, our discovery is very, very significant. So, next, here you can see we have collected ample amount of winged fruits of Engelhardia from Pliocene sediments of Jharkhand, Eastern India. Here I am showing you the various outcrops from where I have collected ample amount of winged fruits of Engelhardia. Here you can see this is the outcrop 1, this is outcrop 2, this is outcrop 3, and this is outcrop 4. So next I come to the geology of the fossil locality. So earlier workers have considered the age of this formation to be late Tertiary based on the study of megafossils as well as palynological data. However, no absolute radiometric dating has been carried out yet. But Prakash et al. In 1987 assigned the age of this sedimentary deposit as a Pliocene based on the presence of Sindora wood, as Sindora is an index fossil of Pliocene age. Next, I come to the lithology of the fossil locality. Lithologically, the sedimentary unit is composed of sandstone clay shell beds. Conglomerate occurs in extreme beds of the sedimentary outlier. The unit overlies the granite gneiss basement of Birha and Rampur Nala rivers. Next, I come to here you can see these are the ample occurrences of various types of trilobed winged fruits of Engelhardia, which I am going to describe in latter part of the slides, each one of them. Next, here I have presented a schematic or a diagrammatic representation of the trilobed winged fruit. Here you can see this is the median lobe and these are known as the lateral lobe. The median lobe shows a pinnate primary venations followed by secondary venations of brachiodermis in nature, that is forming a loop near the margin, quad tertiary veins forming a reticulate maze, and the size of the fruit ranges from 4 to 7 centimeter, having an entire margin. The fruit bears a nut. The nuts are bilobed in nature. Sometimes the nut is covered by hairs also. So next I come to the, here you see that we have recently published an article, the first fossil evidence of Paleocarya from India and its biogeographic implication in the Journal of Systematics and Evolution based on our earlier field survey. So here you can see this is the fossil fruit of Engelhardia showing hairy structure of the nut. These are the wing and these are the median wing, these are the lateral lobe, and here you can see a large view of the nut. So next, here I begin the each description of each one of the fossil wing fruit which we have recovered recently. So here this is the type 1. Here you can see this fruit is characterized by an acute apex. The median lobe is oblanceolate in shape, having nearly about 50 millimeter long and 10 mm wide, showing, however, its lateral lobe is incomplete. Next, the venation pattern is secondary venation pattern is brachiodermis and it bears 9 to 10 pairs of secondary veins which diverge at an angle of nearly 40 to 50 degree. Here one more interesting thing is that the mid vein and ends near the basal region you can see these are the 3 primaries aimed at the basal part of the fruit. Next I am going to describe the leaf remains. We have also recovered leaf remains along with the fruit. Here you can see this is the leaf of Engelhardia, and last we have shown the modern leaf of Engelhardia, which is similar to the fossil leaf specimen. The fossil leaf is characterized by a elliptic shape, approximately 20 centimeter long and 4 centimeter width. Petiole is very short. Here you can see this is the petiole. Margin is entire. Apex is acute. Base is obtuse. Primary venation is pinnate. Secondary venation is brachiodermatous, and the angle of divergence of secondary vein is approximately 60 degrees. Next, I am going to describe another type of winged fruit which we have recovered from the same sedimentary unit. Here you can see this fruit is characterized by a long median lobe, and one interesting feature of this fruit is that here you can see this fruit, the nut is covered by a hair. Earlier the earlier fruit does not show the hair. Here you can see this nut is covered by a hair. Next, another part. This is also another type of fruit which we have recovered from the same sediment. Here you can see this is also incomplete fruit. Here you can see the apex part is round, and here it shows this brachiodermatous nature of secondary veins, and the main vein ascends showing 3 primaries. Next, this is another type. This fruit is also incomplete. It is showing only the mid vein. Here the apex part is almost round, and it bears nearly about 10 to 12 secondary venations, and the nut is almost like a disc. Next, here you can see this is also another type of Engelhardia fruit. This is characterized by almost acute to round apex, oblanceolate in shape, and the size is almost 55 millimeter long and wide is almost 8 millimeter. The lateral lobes are not preserved. Midvein is extending to the lobe apex. Secondary venation is brachiodermatous, diverging at an angle of 40 to 50 degrees. Tertiary veins are reticulate, forming a mace-like structure. However, nut is not preserved in the specimen. Next, here you can see this is another type of fruit which is showing only the median vein, only the median lobe, other 2 lobes are not well preserved. Median lobe is very long, oblanceolate in shape, almost 65 millimeter long, and the wide is 9 mm. And the lateral lobe is incomplete. Mid vein extends to the lobe apex, and the secondary veins are 10 to 12 in pairs, diverging from the main vein at an angle of 50 to 60 degrees. Tertiary veins are finely visible, and the quaternary veins are not clearly visible. Nut is also not preserved. So next, I come to the biogeographic implications of the fossil Engelhardia. Presently, as the genus is distributed in tropical forest of Southeast Asia. However, based on the earlier fossil record, the genus was established during Eocene in North America, followed by post-Eocene from Europe and Southeast Asia. Thus, judging by the fossil record of Engelhardia, we can say that the Engelhardia might reached Europe from North America and finally colonized in China. Then it became widespread during Miocene and Pliocene. Here you can see this is the biogeographic map of Engelhardia that shows past as well as present distribution. This green color represents the present distribution where Engelhardia is presently growing, and here you can these white dots are showing the past fossil record of Engelhardia. Next, I come to the distribution or the possible migration routes of Engelhardia. Engelhardia reveals a disjunct distribution here, which suggests that migration via various land bridges might be the reason for widespread. Engelhardia, here you can see this is the biogeographic, this is the migration route. Here you can see this, the Engelhardia might have reached Southeast Asia from North America via BLB, that is Bering Land Bridge, and the Engelhardia might have reached Europe from North America via NALB. Here you can see. Next, I am going to discuss the main or the possible reason for Engelhardia from the fossil locality. It is interesting that Engelhardia no longer exists in the present day flora of Chotanagpur Plateau, but now indigenous to eastern Himalaya. We assume that the extinction of Engelhardia might be due to climate change. During Neogene, different environmental change in Asia took place due to orographic development as well as increasing aridity and seasonality in late Miocene and Pliocene led to gradual disappearance of Engelhardia as it could not adapt to the new environment and suffered local extinction and finally shifted to suitable areas with more congenial ecoclimatic conditions. Other significant reasons might be the monsoon intensification as well as the climate change since Pliocene. So it could not adapt to the recent the changed climate. So it shifted to the new environment. Next, I owe my deepest gratitude to my guide and supervisor, Dr. Mohsen Ali Khan of Department of Botany, SKU, and Professor Subi Vera, University of Calcutta, for suggesting the research problem and providing constant guidance during my work throughout from the beginning. I'm also indebted to other co-authors for their constant support. My sincere thanks are due to authorities of BSIP for providing the platform to present my work. Here are some of the key references. Here you can see. And last, thank you all. Thank you, everyone.
Thank you very much for this nice presentation and this overview on the paleobiography of Engelhardia. Are there any comments from the audience? If this is, there is BSIP Online raised the hand.
Myself, Gaurav. Very good talk, Tapushi.
Good evening,
You
sir.
have presented a very good evening. You have presented very well, and
Thank
you
you,
have
sir.
very good fossil records from the Rajganda formation. Actually, this is I'm not asking any question. I am just worried about the age of the Rajganda formation,
Yes,
which
sir.
is assigned as Pliocene. There
Yes.
is no age. I think there is no definite age
Actually, a
control
definite
for.
yes, sir. Yes, there is no definite age, no radiometric dating has been done yet, but based on the earlier record, that is earlier papers, as well as the report from the Geological Survey of India, they have assigned the age to be Pliocene.
I think the Dr. VP Mishra sir is here from GSI. He will enlighten, say a few words about this.
Yes, yes,
Sir,
sir.
VP Mishra sir.
Rajganda, I initially worked way back in 1976, 77, and 78, long back.
Yes, yes, sir.
What initially we thought that it's upper tertiary, but
Yes, yes.
it seems that it should be designated as either late Pliocene or maybe somewhere in the Pleistocene. I am not very much sure. Yes, radiometric dating has not been done, but in addition to the leaf impressions which we described with Dr. GP, Dr. Bande, and so many others from BSIP, we did record fossil wood Sindora.
Yes, yes, sir.
So
Yes, sir.
the beds of Sindora are a bit older than the sales from which you are getting the fossil leaves. So it's very difficult to say, but probably these should go in the late Pliocene, it seems.
Sindora fossil wood is also known from the Miocene sediment.
But those are below the beds which had, from where these Sindora fossil wood fragments were collected, are older to the sales. And these sales are just overlying it, and typical unconformity are likewise. And I visited long back, way back in 1976, 77. So the big gap is there, and only BSIP has been working on these, and these need specific dating. In fact, recently, Dr. Bera has also come from Calcutta University, perhaps. He has also come out with a paper on fishes from there. He also collected.
Yes, sir.
It's interesting collection. Congratulations to you.
Thank you, sir.
Thank you for this discussion on the unresolved age, which in many cases we have this problem with plant fossils. Bob, would you like to raise a short question? Maybe?
Yeah, Taposhi, well done. Very
Good
clear.
evening, sir.
Good. presentation. You reintroduced me to an old friend. I found Engelhardia in a clay pit in Tennessee with David Dilcher in 1972. That was my first introduction to Engelhardia. My question was about the dating as well. In one of your slides and in the papers that we have written together, you reference the fact that there are pyroclastic deposits nearby. I, as you know, have never been there in the field, but potentially it should be easy to get some radiometric dates from this material if you understand the relationship between the pyroclastic deposits and the leaf-bearing horizons. It's a very simple thing to do, and within a matter of a few months, if you use argon, you can have a very precise date. So I would urge you to go back in the field, look at the relationship, collect the pyroclastic flows, and let's deal with this dating issue once and for all.
Okay, sir. Then in the next field, we will try to find out the pyroclastic rock, and next we will try to get the approximate dating.
Yeah. Thank you.
Thank you, sir.
Thank you very much.
Thank you,
you,
ma'am.
Taposhi,
Thank you, ma'am.
for this presentation. And now we are coming to the last presentation of today, which is on new legume fossil leaves from the Pleistocene of Nepal, presented by Dan Bahadur Khatri. Yes, he is there. Please try to share your screen. And Taposhi, please unmute, mute your mic. Thank you. So let's see what's happening now. The last try.
Entire screen?
Not yet.
Yeah. It's here. Why it's not working?
Yeah, now something is coming.
I think it's working. It is coming.
Not yet. I don't know if you have a feedback again on your screen. Oh, it's frozen, isn't it?
It's working here, but I don't know why it's not working.
It's presenting now.
You are sharing your browser? No.
Does it appear now?
Yes. Yes,
Is
it
it
looks
okay?
good. Yeah, please go ahead.
Namaste and good evening, everyone, and good afternoon to all of the researchers and the persons who are working on different field in the research purpose. Here, the two legume fossils leaves from Pleistocene sediments of Nepal has been reported for the first time. Outlines are categorized into 4 parts: introduction, materials and methodology, results and discussion, and finally conclusions. And firstly, the whole Himalaya is divided into the screen is working. I'm sorry, the structural divisions, it's not working. Why? The study area in the Google map, it shows that the location lies in the central part of the Himalayan range. The two fossil localities were observed from the Kathmandu Basin. One is from the bulk of the Ksinkali road section from Bhutkhel, and another one from the southern part, the Naphu Farsidol road section, Bungmati. And this is the geological map of Nepal, where the Kathmandu Basin, the Intermountain Basin lies. And this is the geological map of the study area, where the older section, the Lukundal Formation, and the younger section, the Gokarna Formation, from where the two fossils were obtained. And we use the stratigraphy from Yoshida and Igarashi 1984 and Yoshida and Gautam 1988. And the fossils were extracted from the two different horizons. This is from the younger section. This is from the older section, and this fossils locality is from the young section. Is from late Pleistocene, and this is from early Pliocene to middle Pleistocene. And the photographs were taken by CyberSat digital camera, and the study of the different vanishing patterns were studied through the stereo microscope. And these are the different fossils that were obtained from the younger section. It's from late Pleistocene, and the lithologic profile is constructed here, which is of the fluvial deltaic to lacustrine sediments. And after observation morphologically to the fossils, the characters were compared with the nearest living relatives, and almost all those fossils that were found from the younger Gokarna Formation belongs to the Alvigia genus. And these are the modern taxa that were compared. These are the fossils, these are the modern extant genus of Alvigia, where the fossils that were collected were compared. And the current fossil, these were they were found the similar characteristics with the Alvigia Liebeck. And another fossils that are from Bohinia species that were collected from the older and younger sequence, that is from Lukundal Formation and the Gokarna Formation. Here, these fossils were obtained from the older sequence, that is from the younger sequence, that is from the late Pleistocene, and this is the same columnar section that were constructed. And these whole different fossil specimens that were studied based on morphological characters and that were compared with the nearest living relatives, and it shows all the specimens. This shows closer resemblance to the extant genus Bohinia. And these fossil specimens that are from the older sequence, that is from the late Pliocene to middle Pleistocene. And again, these fossils were compared with the nearest living relatives. This also shows the closer resemblance with the Bohinia species. And this Bohinia extant flora, this is from Nepal, and this is from India. And what are these existing fossils indicate? What are the relationship with the tectonic events? That is the question that arises. Actually, the Kathmandu Intermountain Basin, it lies on the northern part of the Shivalik, and nearly it's also on the northern part of the main boundary thrust. Here, these existing fossils that were found from the Kathmandu Basin, it shows the major advancement of the Paleo Kathmandu Basin lake dimension during the preservation, and the flora may have advanced during the lake dimension expansion on the Pliocene to Pleistocene time. And these existing fossils that therefore were coincide with the major adjustment advance in the Paleo-Kathmandu lake dimension, and maybe it is related with the tectonic activity promoting upflip of the southern sub-Himalaya or the Shivalik zone. And this map, this global map, it shows the distribution of Helvetia and Boinia, and this pink color, it shows the Helvetia distribution in the modern time, and this pink one, and the light blue, it shows the distribution of Boinia species, and this dark red color, it shows both Helvetia and Boinia species together living in the same regions. And this map also shows the different fossiliferous horizons that were depositing the Helvetia and Boinia species. And here the numbers that represents 27, 38, 42, 59. These numbers, it shows the locality of the Helvetia fossils collected, and Boinia species collected were from different regions that were numbered from 1 to 26, 28 to 37, and 39. And these all of the Helvetia and Boinia species were from Oligocene to Miocene to Pleistocene age. And this is the distribution track, paleo distribution track, where the Helvetia and Boinia has been deposited. Their fossiliferous remain in different parts of the in the earth. And the oldest from the Oligocene to Miocene and Eocene to Miocene lies on the southernmost part, the Australian region and the Southern America. Here, Oligocene to Eocene, Oligocene to Miocene Helvetia fossils, Boinia fossils were recorded, and from here, Eocene to Miocene fossils were recorded. But on the northernmost part, most of the fossils has been recorded from Miocene to Pliocene age. And this is the modern vegetation type in Nepal. And this is the horizontal distance taken from the Google Earth. And this shows the elevation from where around 70 meter from the sea level up to more than 4,000 meter were taken. And these are the different photographs that were taken in different time by myself. And every photographs, they shows from grassland to the subalpine conifer forest. And this is from the Indo-Gangetic plain, Shivalik, Lesser Himalaya, and this is from the higher Himalaya and greater than higher Himalaya. And this is from, both are from, this is the lowermost part from the Mustang, Thakkola area, and this is above from to the northern part of the Mustang area. That is further north, that contains mountain grasslands and shrublands. Probably the height of these mountains are more than 4,000 meters. And the example was taken as a Kathmandu Basin, where the forest type, it has from subtropical broadleaf forest to tropical subtropical coniferous forest, because the area within the Kathmandu Valley is Fulchoki, Chandragiri on the southernmost part and Shivapuri on the northernmost part. So this Fulchoki, it consists of the tropical to subtropical conifer forest, whereas this Kathmandu Basin, it has a subtropical broadleaf forest. Probably the Indian tropical monsoon has affected the study area, and finally some conclusions were made. And in the investigation of the physiognomic characters, habit of these two fossil taxa with the extent equivalence, it shows that a warm and humic climate was there during the deposition of the sediments, And the significant expansion of Bauhinia species is due to the prevalence of warm temperate climate to warm humid climate. However, the Elvigia species is due to the warm and humid climate during the deposition of Lugundal and Gokarna formations respectively. And finally, these analysis of these legume plant fossils reflect the migration triggering local short distance dispersal, probably from the Shivalik towards the intermountain Kathmandu Basin. So these are the conclusions. Thank you for your attention. Thank you. Thank you so much.
Thank you very much for this interesting presentation and for your patience to be the last of today. You said these fossils come from Pleistocene sediments.
Probably
so these Ibiza and Bohemia from late Pleistocene, you said.
Yeah.
So this might be strongly influenced by glacial periods.
Yeah.
And short-term strong climatic changes, not so much the long-term from Miocene development, right?
The glacier expansion and the retreat, it also enhanced the encroachment of the Paleo Lake. However, the fossils that were also recorded from the southernmost part of the Kathmandu Valley, it can also suggest that there may be the possible short-term dispersal of the legume fossils, legume plants from southernmost part. Because on the northernmost part from Kathmandu Valley, the Elvigia and Bauhinia fossils are less found, rarely found, because the range is not much more higher than 2,000 meter. Below 2,000 meter, they are found. So probably there is a short dispersal of the extant plants of Bauhinia and Elvigia.
Yes. Do you think they got extinct during the last glacial maximum? Maybe.
Maybe, but still the research is going on. So
it's yes.
Yeah.
Please continue with your work. I'm curious to see more of that.
Thank you. Thank you so much.
Did I miss something? Is there any other comment on that? I do not see any. So with that, actually, I would like to close the session of today. We had a range of interesting talks about biogeography, several about the Kutch Basin, which I learned a lot about today, and which really has a huge potential for combined work on paleobotany and paleontology. Sorry. And yes, I think all the speakers, the discutants, and all the colleagues attending today. And I thank you also for your patience with the technical issues. And I hope to see everybody back tomorrow at the same time, right? For us in Europe, it's 11:30. For India, it's 3 p.m.
8 p.m.
And here in Nepal, it's 8:24.
8?
It's 3:30 in England.
3.
Take lunch.
Okay, we
Angela
just
was talking about tomorrow.
yes, we are meeting tomorrow at 3 p.m. Indian time, right? This is what I wanted to convey. Anyway, thanks to everybody, and thank you for the technical support, and see you tomorrow. Bye.
Bye.
Bye-bye. Bye-bye. Bye.